is fine, I assume. And get the message. Okay, we are live. Great. Yeah, so we had a technical issue. I think it's solved now. So yeah, welcome again, everyone on YouTube, in class. We have a good number of students here in the classroom. So thank you so much for coming. And welcome to the 10th lecture in computer architecture course. I already started the lecture 5 minutes ago, but I have to repeat it because we were not live streaming the lecture. So hopefully we can cover the thing we were missing on YouTube. So yeah, I'm Mohammed Alser, and today I'll be explaining about intelligent genome analysis, which is a very important application where we can leverage the things we learn in computer architecture course, such as efficient algorithm, efficient hardware architecture that we can use to accelerate this important application. Okay, so the agenda for today, I prepared different topics for all of you. So first, I'm sure maybe you have some background on the topic from the high school, but I'm going to refresh all these ideas for you. We will start with the definition of genome analysis, why we need it to be intelligent, and then we will go through how we do this algorithmic wise, how we can perform genome analysis, how we can perform read mapping. And then I will tell you that why the reason why we have this read mapping step as a bottleneck in genome analysis pipeline, and then how we can solve all these bottlenecks, how we can accelerate it, make it more efficient. And then I will show you step by step the things we propose in our group, Sapphire Research Group at ETH Zurich. And towards the end, I will give you our vision about where read mapping is going next. Let's start with genome analysis. Okay, to analyze anything, regardless of whether it is genomic data or something else, the purpose of analyzing is basically to gain more insights and more observations. We don't care about the numbers we get or the results we get, but rather we want to gain more insight that we didn't know before the analysis. And this is true about genome analysis. So if you look to this picture, you may observe something very interesting. What makes all these individuals different? What cause all these phenotypes, traits, hair color, shape color, skin color, and all these properties? So basically that is genome analysis. So our goal is to identify, measure, or compare these genomic features that makes us different. And it could be functional or non-functional genomic part that makes all these differences. So we would like to identify these, know these, such that we can correlate them to our health status and so on. So to start doing genome analysis, first step is to get one of these commercialized kits from the market or from the pharmacy, where you can do it at home. You can take the sample, swab something from your body, send it back to the company. They will do the test for you, a paid service, of course, and they will send you some of these reports. Could be about your health, could be about your origin, which country likely to be your original country, and so on. In the company side, what they will see, they try to isolate many things from the sample you take, such as blood cells, white and red blood cells, plasma, and so on, and they will keep these chromosomes that they will extract from your cells. These chromosomes in a human, there are 23 pairs of chromosomes, one of them coming from the father, the other one coming from the mother. Of course, there are some genetic variations that are not inherited from either parent. We call them somatic mutation or de novo mutation. They exist there as well. Now, our goal is to go deeper inside each chromosome and read the DNA content. If you look to these chromosomes under electron microscope, you will see something similar to this. And this is a real picture for Henrietta Lacks cells. And this has a very interesting story. If you don't know this lady, this is the real picture. This is the actress that represents Henrietta Lacks in the movie. This lady, the doctor take her samples or her cells, and they kept them in the hospital without her consent. She didn't approve anything about that. She kept the cells for very long. The doctor kept the cells for a long time in the hospital, and they started doing research on that. They even enter not the doctor, but the research community later on, they infer the genetic material of her siblings and her relatives. So you can do much more things on the cell or the DNA rather than the things that were intended for. If you didn't watch the movie, I invite everyone to watch this movie. It's a bit, I would say myself, I don't know, I would say boring a little bit, but the facts are still there. So they didn't do a good job in the movie, but that is, again, my personal opinion. You might find it interesting. Anyway, so once you get these chromosomes, you extract the genetic material from the chromosomes, you get what we call a DNA sequence. And that DNA sequence is, you can consider it as a cookbook. It has all the recipes that makes us. And the DNA, normally it's transcripted into RNA. The RNA is a little bit slightly less stable than the DNA because of the chemical bonds there. Later on, I'm going to explain that. But this RNA is going to translate into protein. These proteins, what makes us? What makes our color, hair color, eyes, and so on. So now back to the DNA. Assume we have a bunch of individuals, 16 individuals over there. Each of them has its own genomic sequence. Now assume we have extra health information about all these individuals, such as blood pressure. Now we have a group of people who have low blood pressure and another group of people who have a high blood pressure. Now, as a bioinformatician, we would like to correlate information from genomic sequences to their health status and try to correlate these such that we can infer what caused this high blood pressure disease. As you are, all of you smart people, you may check here something interesting. We have SNP1, single nucleotide polymorphism, that is single character causing something. And another thing, SNP2. So we would like to correlate these two SNPs to the blood pressure and see which one causing the high blood pressure disease. If you see this, you'll see it's random information, right? We cannot say anything about it. But if you check SNP1, you will see whenever the blood pressure is low, these individuals, all of them have T at that location. But when they have C at that location, then all of them show symptoms of high blood pressure. Then we can infer something about being C in that location, probably they're going to have this disease. They may not have it currently, but they may have it in the future or inherited to their kids. Now, these studies are well known as GWAS, or Genome-Wide Association studies. In all these kind of studies, we need to have two groups of people. The first group, we call it controls. The second group, we call it cases. And the controls, normally people without the subject disease that we are studying. But the second group, all of them, they have that disease that we are currently studying. And we get their genomic sequences for all chromosomes, and we plot what we call Manhattan plot. And you can guess why we call it that name. And here in the x-axis, you can see all the chromosomes of a human, except the 23rd, which is for sex chromosome. And the y-axis, we plot the significance of each SNP, how significant for representing the differences between the controls and the cases. So we set a threshold. Whenever there is a SNP or the frequency of that SNP exceeding that threshold, probably it can tell you something about that disease that we are studying. That is GWAS study where we compare the DNA sequences of two groups of people. There are other studies such as studying the RNA, not the DNA, or correlating RNA plus DNA, which we call transcriptome-wide association studies. If you need more details, you can check this, one of the good papers about the topic. All right, and this is how we practically use it, even in clinical setup. In practice, we have this database, for example, called OpenSNP, single nucleotide polymorphism, where they give you information about the location in your DNA and what genetic variation there and what may have effect on your health status. For example, this is one of the real data you can access using this name. This is the name, this is the chromosome number 19, this is the position in the chromosome number 19, which is this. Now, if you go to that position and you see a patient with a T over there, then he has very low probability to get better if he got this disease. However, if he has C in that location, then the probability is higher to get better or to respond to a treatment for that disease. This is what we call genome-wide association studies, or whole genome sequencing, or personalized medicine, where you give a treatment that is, let's say, tailored to the genetic material here. If you think that disease might be caused by only single genetic variation, that is wrong. The genetic variation can be very large, not just single character in your DNA. For example, this disease, autism, one of them, is caused by a deletion of 500,000 characters in chromosome 16. The same thing with obesity, and that is caused by a deletion of the same amount. However, for underweight, it's duplication. So you can imagine how large these genetic variation to be detected. And that is very challenging for us who designed the algorithm and the hardware architecture to detect these variation. Because after you get this genetic material, it's no longer biology problem, but rather computer science problem, where you have this input data and your goal to design an algorithm to detect these differences. This is one of the best paper. I enjoy reading it about structure variation. It shows all type of structure variation, the definition, the algorithms used to find these. So I recommend everyone to read this paper. All right, hopefully I give you just a brief introduction about the topic, the importance of genome analysis, but what we mean by intelligent genome analysis. All right, so the first aspect in intelligent genome analysis is to be fast. So we want it to be really fast. It could be optimally fast as real time, but normally this is not the case. We cannot have real time analysis such that we process the data as they come from the machine. And here we are talking about the bandwidth, how fast single genome analysis can be. However, also, especially in the pandemic time, we start talking about analyzing everyone in a city and start to see if they have COVID-19 or maybe something else, like whatever the disease is. So you are talking here about the scalability. So how many analyses you can perform per day. It's not about how fast each analysis, but the parallelism, how many of these can be performed in parallel for example, single hospital. The third thing in being intelligent for the genome analysis is to use intelligent architecture. When we talk about population scale, it means we are going to perform this in routine basis, every day, everywhere. So to do that, if the analysis is not energy efficient, then we are going to face a lot of problems, similar when we build a cloud or cluster and so on. Then we need to cool it down, and that limiting the portability aspect. You cannot perform the analysis anywhere. You need to a special place where you transfer the data to that centralized place and then do processing. For that, we need really intelligent architecture, such as small, specialized hardware with less data movement. And when we talk about this, we mean energy efficiency and portability. Also, the DNA, as I told you the story about Henrietta Lacks, is a valuable asset. If you reveal it to someone, then you are revealing information not only about you, but your relatives as well. Imagine if you are sending your DNA to the clinic and you're asking whether I have this disease or not, they can know more about other diseases that you may have, not only the things that you are interested in. And that may block you from health insurance policy, for example, if they know that you have this disease and they are going to suffer a lot, pay a lot of money for that. So it's really important to keep your privacy. Again, when we talk about fast genome analysis, some people or some guys in the community try to propose heuristics such that make the analysis fast. But directly, we ask, how about the accuracy? Okay, you make it 50x faster, but you compromise the accuracy of the analysis. This is not something we want. And all of these aspects leading to what we call intelligent genome analysis. Right, I hope the previous slide or the definition of intelligent genome analysis was clear. Please ask me a question. Please, someone let me know if we have questions on YouTube as well. All right, does it really matter to do this intelligent genome analysis, why not just doing normal genome analysis? I don't care if the analysis takes 1 hour or 10 minutes, right? I can wait. I won't suffer just waiting 1 hour, right? Okay. For single analysis, it might not be that important. But again, when we talk about hospital admitting a lot of patients, all of them need to do such analysis. Or when we are in the pandemic time, we want to do this before the flight for all the passengers, or for all people entering the university, we want to do such analysis, for example. It might not need to analyze the entire DNA in these cases, but still we are talking about scalable analysis, which is very important. There are some other applications that are also life-threatening. It's not about scalability only, but about saving life. And I'm going to tell you some examples about that. The first aspect we were explaining is being very fast. We want something really fast to do the analysis. How many of you watched these two movies, Gattaca and Tomorrowland? This is a bit old. I don't expect few. Okay, we have Bano, all right? We have two. Okay, if you watch these two movies, I'm not sure if the picture here is clear. I cannot see it myself, but probably you can see it. Okay, this is a smartwatch where you can touch it with a finger and it will directly analyze your DNA just in mere seconds and try to identify you based on your DNA. So it's considered as a fingerprint, and if it is you, then to allow you to access these gates. This is a gate over there. You can consider it as an airport, and these people trying to serve there. So this is how we envision it to be. We would like something really in 1 second, 2 seconds, just analyze your DNA, get whatever you want from there, and move on. What are the applications of very fast genome analysis? As I said, think about critically ill infants. These infants admitted to the hospital. There is no clear symptoms. Doctor cannot tell what was wrong because there is no symptoms. The only option they can do is whole genome sequencing. And when you go to any hospital, they will ask you, do you want to pay more or less? If you pay more, you will get analysis in 2 days. If you pay less, you'll get the analysis result in 5 days. That is whole genome sequencing. Now, there are a lot of research, it's already proven clinically, that doing whole genome sequencing for these infants, especially if the symptoms are not clear, then that is going to avoid morbidity, reduce both hospital stay and the inpatient cost. So it's very important to act quickly because these critically ill infants, they have few days to survive. Otherwise, they normally die.
To make sure I understand, this is saying by saving 3 days, you will reduce the cost of treatment by up to $2 million?
No, that is the cost of whole genome sequencing. So compared to not doing whole genome sequencing, try to give them treatment, try if it works out or not. Or comparing to doing whole genome sequencing, compare it to a previously known patient that you already recorded in the history of the hospital, that this patient has this in this location in his genetic material, and we give them a treatment at that time, and he was getting better after 2 days, then probably the current patient, he has similar genetic material, then we are going to give him the same treatment as that patient, and then we are going to cut down the inpatient cost. So that is a whole genome versus non-whole genome sequencing.
Okay, I guess the savings come from both the efficacy of the treatment you now have and also by the fact that you have to do a lot less decision making throughout the course of the treatment.
Exactly. Yeah, that's true. Yes.
And is this per patient, this number?
I think so, yes. They have a table. They show, I think, around 16 patients, and each patient, the price really varies. Depends on what they need. Because they are critically ill infants, what shoots the price is the ICU unit. It's very expensive to keep these kids in the ICU. Yes.
I'm guessing this data was coming from like hospitals in the US.
This study, I think, I forget where it was conducted, but you can check the link over there. I think it's in US, but the next slide is about UK. I think one of these paper in Japan, the other one in US, if I'm not mistaken. But please double check it. You have the link over there. This is the paper, the title, and so on. Yes.
So that's the cost estimated by the hospital, right?
Yes, definitely. This is clinical study, basically, the actual patient, actual treatment, and so on. You have questions? Yeah, I'm happy to take a question. Please ask as much as you want. Yes, please.
And since this is for like the whole DNA sequence, would it be possible to even further improve this if they could cut down on which genome, like which parts to actually look at for these particular elements?
Yeah, definitely. That will reduce first sequencing cost. It's not about patient, but about getting the genetic material to read it. If you are reading the entire genome, which sometimes we call it coverage, so how much you want to cover the DNA, whether you want to cover it a lot or do chromosome-specific sequencing or gene-specific sequencing or single-cell sequencing. So it really varies, the cost of the sequencing plus the time. So you can do, sorry if I didn't define sequencing, but I'm going to define it in the next few slides. When I say sequencing, I mean reading the genetic material, your DNA. So you could read it in 2 days or a few hours or 3 days. This is the norm in sequencing. It depends on how much genomic data you want out of your DNA. Yeah, definitely that is going to change these numbers slightly. But remember, normally these elephants, you cannot get anything about their symptoms. It is critically ill elephants. And for that, you cannot expect certain disease. So you cannot target specific chromosome or aiming to know what was causing that disease or something. So you need to study the entire thing. Yes, please.
You were saying that the sequencing might be done in as little as 3 hours, but I thought, or I guess in the paper that you had written on the overview of DNA's, I guess DNA sequencing, there was, I think you said it was the computational time required for the read alignment was like 32 hours per person. So is that separate from the sequencing that you had just talked about? Yes, that's very good. I'm happy that you read our papers. So when we talk about sequencing, is generating the data. That is the machine-wise. There's no algorithm over there. And when we talk about the 32 hours thing, that is the analysis following the sequencing. So you do sequencing first, you get the genomic data, that is a text format, and then you would like to analyze it using some algorithm or general purpose computer. And when we talk about general purpose analysis, so normally we refer to some specific algorithm on very specific machine. If you change the algorithm, then the numbers is going to reduce or going up, going down, I don't know, depends on the algorithm. If you change the machine as well, that number is going to change. But that is just to give you a perspective or general overview about the workload. Okay, that sounds good. It's just, I guess, last quick thing then, because like the sequencing is really fast, and I think the reason why is because they sort of break the DNA into like several million pieces for each pieces of read, and the reads are, I'm sure you're going to go into this later, but the reads are very short. So you just have a collection, you just have like a big like bag full of like 1 million different sequences of 300 base pairs each. I don't see what you can do with that, but like I don't see how that's useful at all until you go and realign it into like the entire sequence. Excellent. Yeah, I like that. Yeah, I'm going to tell you the full story in the next few slides. Yeah, excellent. Yeah. All right. So this is one of the applications that just tell you it's really important to act quickly and do the analysis really quick. Now let's talk about the scalability. Now still about critical elephants, critically sick elephants. So in UK in 2019, they took the courageous step and they decided to do whole genome sequencing for all admitted elephants. If they are critically ill, directly the first thing to do is whole genome sequencing. So think about any hospital in the UK, they need to have the capability to do such analysis. We don't know about typical day number of patients, but normally you should act to all of them at the same time. It's not fair to have a queue, wait a long time to do the analysis. Think about each analysis may take 2 days. So it could be faster in some hospitals, in some countries. Of course, that is a normal, but on average, we want this analysis to be really quick. Now we talk about the pandemic as well. It's not necessarily COVID-19. This is for Ebola.
where it's happening in very poor countries, very remote areas. We don't have infrastructure to do computation quickly. And you want something portable, like laptop, very small devices, carry over to the environment, try to detect or do testing as much as you can. And most of these equipment were a donation to these countries. So when you send donation, you don't want to wait until you build HPC or cloud computing and all this infrastructure, because simply there's no internet, there's no power, and so on. So it's very important to have this portability feature. This is also a paper, try to use high throughput sequencing. Normally we use a PCR test, right? PCR test is a bit different from what we are talking about here, high throughut sequencing, but this is one of the famous studies, try to do high throughput sequencing for COVID-19. So it still can be an application for that. One difference between high throughput sequencing and PCR, you need to know what you are looking for. So you need to have the strand of the virus, for example, then you are looking for it. But here you can detect many species, given that you can do the comparison between the read and many reference genomes. Some of these terms might not be clear, but I'm going to explain what we mean by reference genome, how we compare to it, and so on. This is one of the studies. If you are worrying about public health, and you are interested in finding what might be harmful bacteria in very crowded places, such as New York City, for example, then one of the studies to do metagenomic analysis or metagenomic taxonomy profiling, we call it. In these kind of studies, we are interested in finding what organisms are present in a given environment and how abundant are they. So this is one of the studies done in 2015 in New York City, where they target 1,500 samples and anything related to subway station. Could be the train door, the ticket machine, the seats, and so on. What they find that about 50% known bacteria, other 50% they don't know anything about, unknown organisms. But they also found something very dangerous. I'm going to discuss in the next two slides. This is a new study this year, was targeting 60 cities. So the previous study was targeting only New York subway stations, but here 60 cities, including Zurich. And in this city, they try to build a map for each of these cities, what kind of bacteria and viruses we have in our environment, where we live. So think about in the next 10 years, we may have a study for the entire world, or maybe in space, try to figure out what we have on Mars, what kind of living, if we have over there, or in different places that we never think about, such as ocean, desert, and so on. So this is the thing that is very dangerous to find in New York subway stations. So they find traces of Yersinia pestis. If you don't know what is this is a bacteria that killed about more than 1/3 of the population in Europe in very few years. This is very dangerous to find in New York subway station. However, this was raising a lot of alarms in the media, but luckily it was a false alarm. It was false positive. And some people call it as a failure of bioinformatics. This is not a failure of bioinformatics. I would call rather a failure of the tool was used for the analysis. And when I told you the accuracy aspect, it's really important to maintain when we want to find intelligent genome analysis. This is one of the important things that we should keep. So we don't want to compromise accuracy just to gain fast analysis. I know when you collect a lot of sample from the city, that is a huge workload. You need to make some hard decision, such as using very fast tool, but maybe the accuracy is still questionable. But this is a disaster if you are telling the public that there's this dangerous bacteria over there and it's not there. Okay, so when we talk also about intelligent architecture, think about the sequencing machine. This is the machine that is going to read your DNA. It sends a lot, outputting a lot of huge data. We cannot call it any more big data. It's really bigger data. It's really a huge amount of data. And we don't know where to process it. Should we use FPGA, GPUs, CPUs, or a new architecture that we are not aware about? We can push for all these new architectures, such as processing in memory, processing using memory, and so on, as Juan was explaining in the last 3-4 lectures. We are envisioning this to be something like that. So this is an portable mobile device, and this is the machine that is going to read your DNA. This is a real device, we already have it, but the accuracy is really low. So we cannot enable such thing currently to be used widely, but this is how we envision it in the future. Something like this, or a smartwatch that you can just touch it, analyze your DNA, give you the information. This is about the privacy. This is the study we did in the past, and we show that what are the things that you can reveal given just the DNA and some information that are publicly available, such as social media. We can know your hair color, we can know your brother, sister hair color, so that we can build their DNA just from these publicly available pieces of information. If you are interested in this, I invite you to read our paper. This is one of the tests that you can do as a privacy-preserving DNA test. They promise to destroy the sample after we send it. They will study only a portion of the DNA that you're interested in. They promise to not study the rest, and so on. So I don't know, this is just one of the examples. Just tell you privacy DNA testing. And now, after all these examples, how to achieve intelligent genome analysis. I hopefully, with the previous slides, I cover the importance of doing it as intelligent genome analysis, but how we do that. Normally, we want to answer such a question: how and where to enable fast, accurate, cheap, privacy-preserving, and exabyte analysis of genomic data. And all the work that we did in our research group, we try to answer that question. And we still yet far from answering that question. It's still an open question. All right, so I can take question now. Yes, please.
The whole privacy thing, it's been confusing to me. It's like, you know, like for computer passwords, it doesn't matter how the different cryptographers are, you know, subway stations, like until you type in your password. And for like your genome, I think it's exactly the same. I really just like go to somebody's desk, just like go to somebody's desk, pick up a piece of their envelope, analyze it, and got their DNA information. Like I feel like it's impossible to control the sideboard channels of DNA information.
That's true. That's definitely true. But there is a difference between someone take it without your consent, or someone is yourself sending the genomic material to the hospital or to the lab to be tested, right? They didn't spend any effort to come and collect these traces after you, right? So there is some differences between you yourself sending this data to somewhere, maybe this third-party place, going to post it online. Could be good intention, like research purposes, could be bad intention, we don't know, but they still can post this information online, right? What you are going to do about that? So there are some mechanisms to stop spreading your DNA and still keeping the research cycle. So they still enable the analysis of these DNA by hiding or altering some of these DNA information, and then still you are able to send your data to somewhere for testing, for health purpose, for research purpose, and so on. So we've done some work in this direction. So I can give you some pointers if you like after the lecture. I didn't post it there, but you can start from the paper I mentioned.
Okay, so it seems like the security aspect of this would be more on preventing leaks on a larger scale. As for like, you know, like if somebody just wants to go and target you though, like
exactly,
then that's sort of out of scope.
Exactly. I can still take your fingerprint, for example, from the table, right? Yeah. You won't do anything about that. But there is a difference between you sending it at larger scale to everyone or over the internet or post it online. Even if you are okay with that, you don't see any harmful thing about sending your DNA to the research purposes to publicly available database, although they are going to anonymize your identity. They are not revealing your name, for example. But that is not okay maybe for your relatives, right? You don't want such cases. Okay, so in theory, yes, please.
Can you go back from to slide TV and to what you offered? I was wondering what do they mean with 330x on genome sequencing versus 100x? Is that the speed or?
Yeah, that is the thing I was explaining, the coverage. This is the coverage. How many times you want me to read every location in your DNA? Why is that? Because this machine, as any other machine, they are not perfect. They impose sequencing errors. They might tell you, you have A at this location, but in fact it's C, right? So to increase the confidence, sometimes you can ask for more accurate sequencing run. That is high coverage. So 100x is normally more expensive than 30x because they are going to run the machine for longer, like 3 days instead of just a few hours, right? But that is going to give you a lot of data, but at the same time, high confidence. When you have overlapping pieces together, you can say something about that. Okay, majority of them in this location, it's A, so it seems it's really A, and that few pieces have it as a C, probably this sequencing error. All right. So in theory, when we would like to analyze our DNA, we want to get something like this. So you send the sample, regardless whatever this sample is, but we want to get the full genomic sequence of your DNA. Unfortunately, we don't have any machine that gives you this as output. Why is that? I'm going to invite you to read these 2 papers. This is 1 and this is 2. By the way, the slides are already uploaded to the course page. Those who are watching us on YouTube, you can access them from the course page. But these are the 2 papers to know why, or what are the reasons why the DNA is cutting down your DNA into pieces and then try to sequence it and give you the genomic material for that. But you can infer that from this picture. This is again for Henrietta Lacks. You can see the scale over there, and this is supercoiled DNA, such as really complex DNA. There is no machine at that scale can directly go into there and start to read character by character. This is one of the challenges. You can think about this problem. I like this analogy. I made it myself. I don't know if it makes sense or not, but this is how I envision it. So these are yarn balls, right? If they are very tangled, very complex, are tweaked everywhere, twisted everywhere, and your grandmother asks you to solve it or untangle this yarn ball to be used for something, how are you going to solve it? One of the easiest ways is to just bring a scissor, try to cut these twisted pieces. Hopefully you can untangle it, right? And this is exactly what we are doing in sequencing. We try to cut them down because they are supercoiled structure, double helix DNA, and we cannot read it using current techniques, current technology. Maybe in the future we have better technology to read it, but currently we cannot do that. And for that, one workaround is to cut them down into pieces, and which is done using sequencing machine. This sequencing machine will give you what we call reads. So each piece outputs from this machine is a read. And for that, so the problem with the sequencing machine introducing a new step that we call read mapping. So if the machine can read the entire sequence of your DNA as one piece, we don't need this step. So this step is only introduced because we cannot read the full DNA. So what we got actually from this machine is chunks of your DNA that are reads, pieces, but the DNA is producing all this amount of data and really quick. So they can give you high throughput data in short time with low cost. Of course, all these numbers depends on what machine you are using and so on. The good thing, the bad thing, the very bad thing, that all these the pieces coming from your DNA are sequenced randomly. So you have no idea about where each piece is coming from. So all of them really randomized, and good luck in finding the right location for each piece. These examples of this sequencing machine, the size really varies. This is a really fridge size. This is a big machine. This is a handheld machine, for example. This is like your PC. This is really a big machine. This is a bit small. This is the smallest, and so on. Now, I'm going to show you some examples about the specs of this machine from 2 prominent companies in this field. The first one is called Oxford Nanopore Sequencing. These are examples of this sequencing machine. This can sequence 1 sample at least, because you can add multiple samples within the same flow cell, but that is a different story. Here, again, this is the same device over there, over in this region. And this is a compute unit attached with it. This is still very small, like iPad size, or maybe smaller, where you can do the analysis over there. Here you can do only sequencing. Here, there are 5 flow cells similar to this. You can do parallelism. Here, again, there are multiple flow cells. And that directly affect the throughput, as you can see here, and the cost. This is one of the companies that produce very long pieces of your DNA, but they are still not the complete sequence. This is another company producing short sequences. And you can see many machines with many throughput, many specs, and so on. But what I want to highlight here, that this is one of the latest machine that has sequencing inside, analysis inside it. So all of them do sequencing, but this is only the guy that can do analysis inside or over cloud. Now I'm going to explain quickly how sequencing is done. And this is about Illumina, the second company I was showing you the machine. What they have a glass flow cell here, glass surface, and we have the DNA strand attached to that surface. And our goal is to know what exact character we have in this DNA strand. We don't know that in advance, and this is our goal. But what we know that we have 4 chemical molecules in our DNA, either A or T or C or G. These are adenine, guanine, thymine, cytosine. These 4 chemical molecules that we are looking for. The A, adenine, is always chemically binded with thymine using 2 hydrogen bonds. The guanine is binded with the C, which is cytosine, using 3 hydrogen bonds. So that is always what we expect. Using these 2 pieces of information, we're going to do the sequencing. So first, we add just T. We don't know what is there, right? We just can sequence 1 base at a time. We add T, and we have an optical sensor that observes if there is a chemical reaction happening or not. If the T was there and we have A, then directly they chemically bind to each other, and that causes a fluorescent light to be emitted, which can be captured by the optical sensor. If we know that we add T and we got some chemical reaction over there, then definitely there was A in that location. And then the machine will produce A. Now, if we add T and nothing happened, then we wash the surface and we add the other molecules. We add A, C, G gradually. Each time we add only single chemical molecules. So we need to add 4 of them to just read 1 base. This is in the worst case. We don't do this for single DNA strand, but we do this with many of them. Actually, it's pools of DNA strands. So that's why we call it high throughput sequencing or second generation sequencing, which is providing very high throughput sequencing data at the time. Now, you can check this video, can explain the things inside the machine if you're interested. Now, with the second company, which we call Nanopore Sequencing, it's totally different story. So we have a nanopore over there that's made out of graphene, for example. We have electrical current passing through the pore, and at the other side, we have whatever sensor can sense the electrical current. And the DNA strand will pass through the hole, and while it's passing over there, it will affect the electrical current going through the pore. It will affect the amplitude or the phase of the electrical current. And then you can observe such changes, and then using some training data set in the past that you already know, for example, all this artificial DNA strand is seized, so you know what effect may have on the electrical current and so on. So I have to connect the laptop. Should have done this previously. Luckily, I have one available. Okay, cool. Okay, once you get this effect on the electrical current, you can infer what are the bases over there based on the distortion that was happening to the signal. I hope this was clear. You can see here, there's no chemical reaction, there's no washing to the surface, and that empowered this technology to read long pieces of your DNA compared to the previous piece, because every time you wash the surface, you are going to destroy the DNA or make it weaker. That's why these kind of technologies are producing longer reads. However, the accuracy is not that high. It depends on how you translate these distorted signal into bases. That's why the accuracy is really very bad compared to the previous one. Yes, please.
Yeah, so I want to ask if one of the reasons why also for the small sequencer, like the Oxford Nanocore, I guess I just want to ask if they also, because of like something to do with the size of the machine, they're not able to break the DNA into smaller fragments, or is breaking the DNA into small fragments actually very easy?
So first, all of them going to break it down. Okay, all of the sequencing machine going to cut your DNA into pieces. Now, why Illumina, for example, cut it into shorter pieces? That is related sometimes to library preparation. And sometimes, you know that the accuracy of this machine normally going very bad towards the end of the read. So normally the last few characters of the read are not accurate. Okay, so if you are going with longer, you are not gaining any benefits anyway. So there is no point of cutting the DNA into longer pieces and then use Illumina devices. That is not going to work as if it is providing longer reads because of this chemical reaction thing. So it's going to weaken the DNA itself.
Okay, so the length of the reads is just tailored to the mechanisms of the technology themselves.
Exactly. And something related to library preparation and so on. I'm not expert in this particular point, but this is what my understanding. Yes.
Library preparation would just be how you choose the chemical reacting molecules, or?
So in library preparation, there are multiple things. One of them is the reagent, the chemical molecules going to add to this DNA strand such that you can process them. For example, in Illumina devices, you build pool out of it because you cannot capture single fluorescent light emitting from single base, but you need a pool of DNA strand that are emitting fluorescent altogether. So you can pick that light. Because as I said, the bases are very small. The scale is really tiny, cannot capture it, cannot see it as just light. But that's why we build pool of this DNA strand, and we replicate the same DNA strand multiple times so that each pool has the same DNA strand. Then all of them will emit light with a wavelength that is good enough to be captured by the optical sensor. Other thing as library preparation, for example, you want to steer the DNA such that you isolate the content of the sample. It could be a red blood cell. You want to isolate them, the plasma, and so on. So you want to steer it at very high speed so that you can quickly isolate all this content and leave the DNA only so that you can extract it later on. So there is a process done before sequencing, actually. It might take a few hours. So sequencing alone is not, might not be the only bottleneck. There are some processes done in the wet lab. When we say wet lab, it means in the actual lab. It's not about computation. All right, if no question. Okay, cool. I don't know if I have a question on YouTube. We cannot see in my screen, but yeah. Okay, moves at once. So as I said, as the name over there, nanopore, because we are using a pore at nanoscale. This is the scale of these pores. It might get smaller over time. I don't know if this is the latest number or not. I think this is a slide I prepared back 2 years ago, but the rest of the slides are already updated. Anyway, what happened to the, I was seeing the picture, right? Are we still online? See? Is the camera on?
No, the camera is not on, but you're online.
Oops.
Okay, now it's fine.
Okay, good thing that I noticed it. Thank you.
Can you a bit turn the camera to the right a little?
I think I'm turning my laptop camera. Yeah, I think I cannot detect the GoPro. Probably it's out of battery or something. It's working? Yeah, I think it's out of battery, probably, right? Oh, what should I do? No, this is my iPhone. Sometimes I use it similar to GoPro. Maybe the cable is not connected correctly. Let's see, new link. Okay, anyway, while Mohammed's fixing it, I'm going to continue because we are out of time a bit. You can see it? No. Yes. Sorry for those on Zoom and YouTube. Yeah, but hopefully you can see me at least, or maybe I can switch it a bit. I hope I won't disconnect anything. Yeah, I hope you can see me now better. Okay. Yeah, so the Nanocore over there is the reason why we call this sequencing technology as Nanocore. And in Nanocore, as I said, this DNA strand pass over there and affect the electrical current, then we can detect these bases. Yeah, there's a video also you can check about these sequencing technologies, so you can get more information and see the real device, how it works. And this is a paper, it was just published 2-3 days ago. It's a very recent one. I'm telling you about machine learning or the application of machine learning, artificial intelligence, and doing base calling for nanopore devices. So as you know, nanopore devices give you a distorted signal as output, and it's not direct, straightforward mapping from electrical amplitude to the bases. You need to really use complex algorithm to understand the effect, whether it was C or G. And there are other challenges such that when you have a bunch of repeated characters, which we call them homopolymorphism characters, for example, all of them Cs. If you see a strand of C or subsequence of Cs or Gs, that is very challenging for the sequencing machine because the DNA just will pass and we will have the same effect on the signal, and you don't know how many bases over there. So you could read it as 4 or 5 bases, but in fact it's 20 bases and so on. So all of these discussed in this paper. I invite you, if you're interested in this or developing some machine learning algorithm for that, to please check it. All right, as I said, common disadvantage with all machines, regardless the price, the specs, whatever other properties, all of them give you pieces of your DNA that you have no information at all about whether which piece comes before, which piece comes second, and so on. So you have no information about their order or which chromosome they come from. And this is another analogy to solve this problem, which is very similar to puzzle solving. So think about the reads over there, and you'd like to build your DNA and try to link these pieces together. For that, as any other puzzle solving problem, you need a reference picture, right? So you need to match these pieces to the picture and then build the puzzle based on that. And this is exactly what we need in read mapping. We need a reference picture and we need these pieces. Okay, now same in the puzzle. If you go to the market, you have always 2 choices. Puzzle with very small pieces with a large quantity, or puzzle with large pieces, small quantity. Which one is better? When we talk about puzzle, of course, this one sounds a better option because we have less pieces, less number of pieces, and for that you have less things to be checked every time you want to put the piece over there, right? However, in sequencing, it's a different story. There is no better. Each of them has different properties. When we talk about short pieces or short reads, then the length is really short, but the accuracy is high. When we talk about long reads, the length is really long, but the accuracy is really bad. These days, the long sequencing technology is getting better. For example, it's no longer 15% in accuracy, but it's something between 4 or 5%. It depends on the base cooler you are using, the sequencing technology. It might go down to 1-2%. That is consensus accuracy. You don't need to worry about the terms. It's out of the context, but just in case you're interested in this, I'm telling you that The sequencing technology here is getting better, but still is not as accurate as we have in short reads. Yes.
Is this the sequencing error or the sort of
Sequencing errors. Yeah. Yes.
How debilitating are these error rates? Can you put them into perspective? Like, is 0.1% error rate enough to make that such that you and I that cannot really be differentiated? 15% making such that I don't know for other one.
Yeah, that's a very valid, good question. So basically, for short reads, you need high coverage, right? So you need to overlap these reads such that you can solve this 1%. Here, we don't use it normally for just single nucleotide polymorphism. We are not looking for a single structure variation or single genetic variation, but rather the application of those are more for when we want to find very large structural variation, very long insertion or deletion. Because they are long, they can close that gap, and then we can know if there's something deleted or not. But these pieces are not good for this kind of application. So each of these has really different application. That's why we cannot answer this question, for example. Which one is best? Which one is better? All of them is needed. However, recently we got a third type of sequencing read. We call high fidelity reads, or hi-fi in short. They were called CCS in the past, like 3 years ago, but now they are called hi-fi reads. These reads normally not as long reads or nanopore reads, but they are somewhere between this and this, closer to be long reads. However, they are very accurate, but they are still expensive to be used widely. So if you want to ask for these reads, you will pay more as if you ask for that read, and so on. So that's why still 3 of them used, because each one of them has their own unique advantage. Some studies, they use a combination of these 3 types, especially as I'm going to show you in one of the studies, very recent studies this year. All right, so hopefully by that I cover everything needed as a background for all of you. And we already spent 1 hour and a half. Now I'm going to, maybe I think good time to take a break. But before the break, this is a very important thing. Any change in the sequencing technology is going to affect all the downstream analysis or the downstream algorithm and hardware architecture. Why that? Because definitely these data are input to whatever algorithm you are using. If the data are different, more errors, for example, then you need to handle that errors first. You need to tolerate it, right? That sometimes we use read polishing tools. Sometimes we use more coverage. Sometimes we need to increase the parameters for the tool. For example, tolerate more edit distance, higher edit distance, higher alignment score, change the penalty for this variation, and so on. So that's why changes in sequencing technologies can render some read mapping algorithms irrelevant. Some of them already did, because there were in the past some sequencing technology before Illumina, and now these days they no longer exist. To get more about the history perspective for all of these sequencing technology, all the read mapping alignment tool and so on, I recommend you this paper, which is coming from our group, recently published in Genome Biology, with collaboration with too many research groups from the world. We were really lucky to have all of them in our study. All right, so looking forward in the future, 5 years from now, 10 years from now, will we be able to read the entire genome sequence as one piece? Is that possible? Think about having new sequencing technique. All of you smart, you can maybe develop one in the future. I think that will deserve a Nobel Prize, I would say. All right, to answer this question, please stay with us until the end of the lecture. Thank you so much. We can take a break now, 10 minutes. So we can come back at 2:35. Thank you. He has a lot of papers. His name is Ivan Eichler, University of Washington.
Okay, sounds good.
But I cannot say if this is the only cause or not.
Okay.
I didn't study this disease, for example.
Yeah. On, I guess, the causality of structural deviations of DNA upon the, I guess, like conditions. Is there like, I guess, phenotypical things you can add to your algorithm to give it higher accuracy?
Yes, there are some algorithms that you combine data, not only the DNA, you can rely on other data to make it more accurate. I would suggest you read the paper I assigned to Banu. She is one of the researchers in our research group. I assigned a similar paper talking about this topic, where you combine multiple data that you can build an algorithm that leverage all these pieces of information. I don't have the paper in mind now, but I assigned it to Banu for reading 2 weeks ago. She can give you this paper if you're interested. No, I didn't put it in the recommended reading because I think you have enough papers to read. I don't expect all of you to read all of them because normally these overview paper give you a lot of information as introduction to the topics. Yeah, sure. Thank you so much. All right, I think we are on time. Oh, someone unmute me. So we're on live. Okay. Anyway, yeah. So welcome again after the break. I think we can start now. I see everyone is back here in the class as well. Okay. So yeah, as I promised, we will answer this question toward the end of the lecture. Now let's move forward quickly. Now we talk about the limitation of sequencing technologies and how they introduce this computational step that we call read mapping. So read mapping is introduced as a reason why we have limitation in sequencing technology. But what is read mapping, in fact? So read mapping is basically we want to map a read to a known reference genome with some minor differences allowed. What does mean allowing some minor differences and why we are allowing for them? As I told you, there are two reasons. First one, sequencing errors. We want to tolerate these. The other reason is genetic variation, which is basically the goal why we are doing this analysis. So we want to tolerate both of them. If we are looking about exact match, then we are not gaining anything. We are building the DNA that is very similar to what we have in the reference genome. How we do that? Simply, we have the sample. We send it to sequencing machine. We got a bunch of reads out of it. And then we have the reference genome over there. And we try to stack each piece to this DNA over there, such that we build the full DNA again. That is similar to the analogy of puzzle solving. Okay, how we do that? First, we need the reference picture to do read mapping, right? And luckily enough, we were able in 2003 to get the first copy of the reference human genome, which was a really big thing. It's really something great to have in that time, but it cost us a lot. It takes 13 years to build and cost for every character we can read, we pay $1. So for a human genome, it's about 3 billion characters. Then the cost for that research, 13 years of research, is $3 billion. It was really collaborative effort between US, UK, Japan, and many more countries, but at least we got that copy. If I told you that was the reference genome, I was wrong, because only this year we could close some gaps in that reference genome. It was really not the complete reference genome, but only this year we could add up 238 million new characters to that reference genome so that we can have near complete reference genome. So only this year, after 3 decades of research, we could get something near complete. But also that one we got in 2003, it was really good enough for doing some applications. It's getting updated almost 4 times a year. So it's not single version. We have multiple versions of the human genome, but at the end, it's only one reference genome that we are using for all type of research. This is still not complete because, for example, the Y chromosome is not complete over there as well. There are a few gaps as well. So we still are not having the complete reference genome. That is near complete. Yes.
You call it the reference genome, but is it like someone's genome, or where does it come from?
It's an assembly of many genomes, but at the end represent one, or at least very limited population of people. So it's not representing the entire world. And this is, again, another problem that I'm going to touch upon in the next few slides. Thanks for the question. Yeah, so how we can get this reference genome? It's publicly available on NCBI database from NIH. You can access this link. You will get a file similar to this. This real lines from the file, the first few lines. The first line is the ID of the reference genome, and the rest are the content of the DNA. You can see the first few characters as N. It means not defined, so sequencing machine cannot read this location. But I think after 1,500 characters, you start to see a lot of ACGTs. Okay, so how long the DNA is normally? For viruses, a few thousand. For bacteria, few million. For human, 3 billion. And we still are not the best in terms of DNA length. So red onion has 5x longer DNA than ours. This Japanese flower has 50x longer DNA than ours. So it's really varies the workload. That's why when we said analysis time or sequencing time, depends what you are sequencing, right? You can see the workload's really different. Okay, now we assume we got the reference genome. How about the reads from the machine? They are also available online. So you don't need to do the sequencing or pay money to get these data. You can access one of them from here, from SRA database from NCBI. There are also ENA from UK. They are almost have the similar data set here and there. But this is one of them, one example you can access using this link. You can see information about the data itself, what machine was used to do the sequencing, the run itself, the length, and many more about the data itself. So both of them publicly available. Good luck with designing your own algorithm. Okay, now let's learn and practice how we do mapping a read. One option is to do a brute force where you check every single location in the reference genome. So you get the read, you check every location in the reference genome, and you see where it fits. Then you repeat the thing for the next read, for the third read, until you finish from the read set. That is a brute force. And as you can guess, it's really expensive. The time complexity is huge. You cannot afford that. Okay, in our paper again, we explain too many things about the read mapping algorithms. What are the available options that you can follow to not use the brute force approach? And we got a very good feedback from the community after we published our papers. They were appreciating the work, so I recommend everyone to read the paper, and we thank the community for their helpful feedback. Okay, so to map a read in practice, without using the brute force inefficient algorithm, we want to have it, or how I envision it myself, it's really similar to querying the yellow pages. For those who don't know what's the yellow pages, is this guy, where we use it as a dictionary for the phone number. So I was using it in the 90s. I'm not that old, I'm just 30 plus something, 33 years old. But if you don't know it, you can use exactly same example as Oxford Dictionary, for example. If you want to look up a word in the dictionary, what you do, you go to the first few pages where you have the index, and then you check the first letter of that word. If it is A, then it will tell you go to these this chapter or these page numbers, right? Then you go there, you check the second character in the word, and then the third, the fourth, until you get the full word, and then you get the meaning. This is exactly the same thing in the phone number. If you are looking for phone number of someone, we were using this book to fetch that number. What you do basically, you go to the index of the yellow pages, and then you use the first few characters from the DNA strand or from the name, and then you try to see what chapter in the book this DNA strand exists. And then you retrieve these pages of the chapter, and then you try to match the rest of the DNA strand until you get the location, the reference genome. And this is exactly how we do it for genomic example. How is that? So here, an example of the reference genome. Here's an example of the read set. So the reference genome has 2 lines. The first line, the ID. The rest of the lines are the genomic sequence itself of the human genome. The FASTQ file has 4 lines per sequence. Every read has 4 lines. So if you have 10 reads in your file, then you need 40 lines. The first line is the ID. The second line is the read itself. This separator has no meaning. And this is the quality of the sequencing. So every base in the read has a quality, or the confidence of the sequencing machine. This is an ASCII code, so you can convert this into ASCII, and then you got the ratio of the confidence, whether this is something to rely on or not. Anyway, how we do now read mapping? So as I told you about the yellow pages, we need to extract information from here and build the index out of it. So we extract randomly or following a method where we pick these pieces from the reference genome, not from the read set, and we build the index on top of these pieces only. We don't use the entire reference genome, just these pieces. Now assume we have the index structure, regardless whether it is lookup table or hash table or any kind of indexing. Now we want to use that index. So we go to the read, and we collect pieces as well from the read. Remember, the read is only the second line, not the entire file. Second line every 4 lines. Now we get these pieces, and it's the time to check the hash table or the lookup table and see if it exists or not. So we check the hash table, and we get the location of all blue pieces, the location of all green pieces, and the location of all red pieces. Now we need to make sense out of all these information. We got a lot of locations, and we need now to examine them in a certain order. In the read, the blue piece is followed by green, and the green is followed by a red piece, right? So we are looking for something similar in the reference genome. So we sort the location of all these 3 pieces when we find them in the reference genome, and we use them in the same order. So we want the blue, followed by the green, followed by the red. And once we do that, we get insights that it only exists in this line, somewhere around these 3 pieces, because only in this line we have the same order as we have here. I hope this is clear. Now the time is to compare the entire line over there with whatever we have here, try to infer what differences, what similarities we have. Now there are some other challenges. For example, we got many lines matching these 3 pieces, right? Because these pieces are short. That is also true. That is a challenge for read mapping. Okay, how we do this again, using algorithms. So we have the reference genome, we extract pieces, we build the index, and in our paper, we find that the hashing is the most popular indexing techniques since 3 decades. So use hash table as one example. We store the seed itself or the hash value of the seed, and we store the location where it appears first. For example, location 1 means the seed appears at location 1, but it also ends at 1 plus k minus 1, for example. That is the end location of the seed. What is k is the length of the seed. All right, so now we build the index. We call each piece of these as a seed or k-mer, where k is the length of this piece. And when we say the length is 6, then we call this 6-mer, or 5-mer, or 9-mer, and so on. So k is really the length of the seed. Now, this is just to give you an insight about how heavy this workload could be. You can see the index size, or the size of the hash table, plus the reference genome. The reference genome originally is 3.2 gigabyte if you consider each character represented using 1 byte. So it depends on how you represent it, because these are character text file, right? So if you represent each character using 1 byte, then it's 3.2 gigabyte. If you represent using 2 bytes or 2 bits, that will change the story. But this is how the original file. And this is the index size. This is the indexing time need to build the index. So you can see it really varies from 3 minutes all the way to 1 hour. Why is that? This is the main reason. So it really depends how you extract these seeds, how many seeds you extract, how long these seeds. So that will change directly the hash table. So it can be overlapping, non-overlapping, space, adjacent, and so on. So you can control that. These are different indexing techniques, starting from hashing, Kurzweiler transformation with the FM index, or suffix tree, and so on. So you can see the time and the memory needed for that. For more details, please read the paper, but you can see the trend. This is not related to the years. So the recent tools, for example, even hashing, they are somewhere here. They are really now more efficient. In Kurzweiler transformation, you can see the trend. They are always efficient because this is a compressed index. It's not as in hash table. But again, it has its own limitation, as we discussed in the paper. Now, assume we build the index. We have the reads. Read number 1. We extract seed from the read, and we use the hash table to see if they exist or not. And we sort the location, as I said. And the read one, only it makes sense when we start with location 5, 7, and 9, because the yellow is followed by blue, followed by green piece, as we have exactly here. So we start with location 5 in the reference genome, and we fetch the next 100 characters, for example, if the read length is 100. If the read length is million, we fetch starting from location 5 all the way to million 5 minus 1. And then we do what we call sequence alignment or verification step. Why we need this step? This is only between the read and the subsequence of the reference genome that we fetch, that we just extract from the reference genome. And we do this step to infer where are the exact location of the differences and the matches. So this is something, use dynamic programming table. And towards the end, it will tell you that there are, for example, 16 differences between these. And this 16 represent the optimal number of differences. So you cannot find something less than 16, for example. And then you do what we call backtracing through this table to find the location of these differences, 16 differences. Then we report them in this report, which we call it SAM format. If this is compressed, then it will be BAM, binary representation of the SAM format, and that will be the output of read mapping. This file is normally a multi-column format, so it has about 12 to 16, depends on the tool. Each column has a meaning, but what is important in these columns is the number of differences, where are they, and so on. It has also information about what read this for and the location in the reference genome. Okay, what do we mean by sequence alignment or dynamic programming table? One way to do it is to calculate edit distance, which is defined as the minimum number of edits that could be insertion, deletion, or substitution. Need to make one sequence exactly look at as the other sequence. So you have two sequences. You want to make one exactly look exactly as the other. What you should do, you may need to do some changes to the first sequence so that it looks as the other one, right? I have an example. For example, organization and operation. You want to make organization look exactly same as operation. What you should do, you can do it using one of these two ways. Again, we are looking for the minimum number of edits. You could do it using more edits, here 7, but you could do it using 10 or 15, but we are always looking for the optimal number of edits. What are these colors? Here insertion, here deletion, and the yellow is a match. So you need to insert 2 characters, delete all these characters so that the entire thing over there will be exactly the same as there. You can alternate over here, for example, and you have 2 ways now to make this similar to that. Now, both of them have the same number of edit distance, that is 7. Which one is better? It depends on you, the user. He will specify a penalty for insertion, a penalty for deletion, a penalty for substitution, and also a penalty for the gap or the deletion. So you want longer gap or shorter 2 gaps, for example. That is also user configuration. So you are telling which one of this is better for your case or the disease you are studying. That's what we call alignment score or scoring scheme. This is another example between organization and translation. There are different ways, but again, as I said, we are always looking for the optimal one, which is here has 4, but I think here has 5 changes. Okay, One example of sequence alignment is called Smith-Waterman algorithm, and it was the most popular since 3 decades. However, also Hamming distance is the second most popular. This is dynamic programming, but this is very simple algorithm. How many of you know Hamming distance? Okay, great. Yeah, this is an algorithm proposed by Richard Hamming, who's a scientist who has the saying in the first slide, if you remember, about data analysis. And the goal of this algorithm was proposed for encoding data for transmission or for communication, where you do error correction over there. And how you do it, you just perform XOR between two sequences, right? Just XOR between every sequence and the corresponding sequence from the other guy or other sequence. And the number of differences or how many ones you get out of the XOR operation, that tell you the Hamming distance. If you got 6 ones out of this sequence, then that is a Hamming distance of 6. Now, as you can tell, the Hamming distance is just XOR, so they cannot detect insertion or deletion. They are only able to detect substitution if you change one character to another or not. So that's why this has own application. This has its own application, especially when you are looking for single variation, not for complex variation. This is one of the tools that my PhD advisor proposed back in 2009, John Alakan. So here he is doing full read mapping using a hash table. If you are interested in this, please check this. The tool name is called Mr. Fast. The code is already publicly available. It's very easy to read. Interested to learning the basic on this. However, it's not the fastest, as you can tell. It's proposed in 2009 and now almost 12 years. There are more mature tool or fast tools such as Minimap2, which is one of the state of the art. And it's also easy to read. I enjoy hacking the code and changing it. You can see the memory footprint of it. It's not the best, but the time is always the best over there. So there are other tools that might do better than Minimap2. For example, these tools, they have almost the same as execution time as this, but the memory is better than Minimap2. However, again, this depends on the data set, on the configuration or the parameters used for the tool. Yes?
What is the variation in the parameter? Like, do we have to give the same execution time and memory?
That all of them use same reference genome and same data set. We have multiple data sets, so each point here represent different data set, but almost they have the same workload. So we usually think 12 data set in this paper. So you can see 12 points for each of these plot. The variations depends really. For example, here you can see a huge difference because sometimes some reads are easy to detect. Sometimes some reads are really complex to detect and so on. It depends, for example, the similarity and the differences between the read and the detected subsequence in the reference genome. It might have a lot of variation so that you need to do more job in the sequence alignment, or it could be just exact match where you can easily track these things. And also depends on the algorithm, of course. However, one interesting thing to note here, you can see most of the tools proposed in the last 10 years or so, they are always have almost the same execution time. Why is that? This can give you some insight about the following. So this is a plot showing the cost per genome for sequencing only, without the analysis, over the years. You can see in the last few years, we start to have sequencing much faster than the Moore's law, which used for the analysis. So Moore's law, expecting that the number of transistors is going to double every some few months, 12 months or year and a half, and that will directly affect the analysis, right? However, for sequencing, it's getting way faster as a throughput to generate data for you to be analyzed. And that is happening towards after 2013 or so, where we start to have the nanopore sequencing technology. At this time, we start to have Illumina. It was the most prominent one for a very long time until we got nanopore over there. Then the throughput becomes higher because we have longer reads now. And then at that point of time, we realized that we really need something fast because now sequencing machine can generate a huge amount of data that we cannot cope with them. And you can see the trend when you combine all performance of the data proposed after 2013. You can see that as a trend. All of them become faster. But the tools in the past, they were randomly. They could be as fast as now or slower, because we did not realize the need for faster read mapping. Yes.
For this extremely, or I guess for this, extreme improvement in sequencing speed, is that also at the cost, or is that because of the introduction of it's called the Oxford Nanopore type sequencing? Because in which case, I'm not sure if this graph, would be it'd be a bit confusing then if before it was slow and accurate, and now it's really fast, but at the cost of being inaccurate. So is that the case, that as time progresses, it's gotten faster, but also as a trend, a lot more inaccurate?
Yeah, that's a very good question. However, in this plot, we did not split it into Illumina-like devices and Nanopore-like devices, right? So this plot representing what's the cost per genome. So it could be for both of them, even at this point. So this is not only for Nanopore, that is still including Illumina over there. However, at this point, we start to get long reads, right? Illumina is also there, and Illumina is going through a large advancement. Nanopore as well. They have high parallelism these days and reducing the cost because they can integrate more flow cells within the same device, so they can process more samples within the same device. So that could be because we got long reads. It could be also because the machines themselves getting better over time, especially for Illumina. These days are having very high throughput because they can process more pools of DNA strands. They can generate high data, plus the cost is going down for all of the sequencing technologies. So all of these reasons together, they start to give you more data. And we realize, it seems from the analysis of the data performance, the tool performance, we realize that we need to be faster at this point of time.
Oh, wait, yeah, I guess what I was trying to ask then is just, is that plot properly normalized for the accuracy of genomic sequencing as well?
No, that is only the cost, but you can guess it. When you use Nanopore, of course, the accuracy of sequencing, it's not the accuracy of the interpretation. That is two different questions, right? The sequencing accuracy might still bad, but you can still do good job in the analysis because there are algorithms can do polishing and error correction. You can correct these sequencing errors, especially if you need high coverage data. So you can do high accurate interpretation, although the sequencing error is there. But that is an interesting question to check the accuracy of these. All right, that was all for read mapping. However, there's more interesting problem or more challenging than read mapping. There's still use read mapping, which is metagenomic analysis. The things I was explaining about New York subway stations. When we have a sample, it's not single individual. It's not single person in the sample because I just swap it from a city or train station or from anything that I expect to have bacteria, viruses, and many more, right? So I send the entire sample to the sequencing machine, and I get reads out of this sample. Now, since there is no single individual that I expect, I cannot compare to single reference genome. I need to compare with all of these guys, which is a database of known species or organisms. So I have a reference genome for viruses, for bacteria, for human, for plants, for many more. Depends on your study, what you are looking for. And then I start do read mapping with all these reads with all these reference genome. Again, you can see here, part of the reads map to this reference genome, other parts also map to this, because it's very challenging. When we compare short piece with long pieces, you may find many places where it mapped to. And that's what we call metagenomic analysis. This is something like, this is where we do sequencing or analysis for single human, but here where we do metagenomics. So you can see all these M&Ms. If you're interested in this topic, please check the papers. This is 1, 2, and 3 about this, coming from the things I was doing at UCLA with our collaborators. Okay, what are the challenges in read mapping? So first, we need to find many mappings of each read. So whenever it's mapped to any location, we need to find all these locations. That is one. So it's not the first good, for example, first good location. We need to tolerate variation, sequencing errors in each read. So we need to use complex algorithms such as sequence alignment. It's not like just Hamming distance, XOR, but rather we need to build this dynamic programming tables such that we can find the optimal arrangement of all these variation. We need to map each read very fast because that will affect the overall performance. And we need to map the reads to both forward and reverse strand. So I got this question in the break. Do we sequence both strands or one strand? Basically, we sequence single strand. And our DNA is double helix structure. We have one strand, and we have the reverse complement of that strand. This is how our DNA looked like. And there are here hydrogen bonds between these two strands. Now, when we do sequencing only for one of them. So the data you got is only for one of them. Now, to do the analysis, you need to imagine that you have the other piece, which is the other strand. We call this forward and this reverse complement or reverse strand. When you do the indexing, you always need to index the seed coming from here plus its reverse complement. And when you get the pieces from the read, you check the hash table regardless if it's coming from this strand or the other strand, and you check if it's similar to reverse strand, you need to build up this and you extract the subsequence from here, not from there, because simply the read map over there. If you're given one of these two strands, you can directly build the other strand because just simply reverse complement. For example, here is T, this is always A. If this is G, this is always C over there, and so on. So that is maybe more details, but just because your colleague asked about it, so I'm mentioning it. Yes, a question? Okay. Yeah, and that raised another question. I always told you that we are using one reference genome, but what if we have more reference genomes for a human? Because European, apparently different from African, different from American, different from Asian, even in genetic material, right? So what we should do about that, how we do read mapping. So in this study, for example, it shows that African has at least 10% more DNA than others. So it's not fair at all to do read mapping for African individual to use that reference genome. We need really population representation of each city, for example, or a group of people where we have their own reference genome where we can do these analyses. And this raised the question or the field of research that called pan-genomics. In pan-genomics, we are still targeting single individual, which is a human, for example, but we differentiate between each cluster of people. So each group of people, we have their own reference genome, and then we do read mapping to that reference genome. How we represent that? So you know that since we are targeting a human and most of the DNA is similar between all the humans, then the best way to represent many reference genome is to use a graph. So we use a graph where single path represent similarity between multiple reference genome, but when we have branches, it means they now start to be different. That is a very efficient way to represent multiple reference genome. Yes?
I'm not sure I understand this correctly. So this means that one genome can have different numbers of bases in different people.
Definitely, because there are duplication, deletions that make it shorter, longer.
Yeah, that makes sense. But the number we saw in the beginning, so the position where the S and P's, I think it was, occurred, don't they change in that case?
Definitely, that will change. But again, if you have insertion or deletion, it means you have the disease, right? Then it depends on what you are looking for. If you are comparing with that disease, you should expect to have insertion or deletion.
So the position in that case is always relative to the reference genomes in that case, right?
Yes, should be. Okay. Should be. Yes, it makes sense. Mm-hmm.
what magnitude of improvement then can you get if you find a more appropriate reference genome?
Yeah, that is a very good question. I cannot answer that. I don't know. For that, you need such studies, right? Pangenomics. So you may need to repeat one of the previously done experiment and then compare it with when you have multiple reference genome. So we already do the studies compared to single reference genome. So we don't know if the results were accurate or not, but when we now have multiple reference genomes and you are considering a single population that you know that all of them, for example, European and stay dead for a bit of time, they didn't move to Africa or they, I don't know, whatever effect they have from environment is not there, then you can compare with the previously done study and you can see if the result they are different or not. But that field called pangenomics, I didn't work in these kind of problems before, so I cannot comment really on this question.
Yeah, just make sure I was asking the question clearly. I was talking about the amount of time, the amount of time required to do the.
Okay, time, not accuracy.
Yeah.
Yeah, for time, I would say wouldn't be different if you change the reference genome, because it could be, again, relevant to the amount of workload. How long is the DNA, right? If it is real long, if it is really short, then that will affect the indexing time, the read mapping time, and so on.
So I guess, like, is there no correlation between the edit distance and the amount of time required for read mapping?
It will affect definitely, especially the last step, sequence alignment or verification. When you use more edit distance or higher edit distance, that will result in having longer execution time for sequence alignment. But for if you mean the difference between linear representation of reference genome and the graph representation of reference genomes, definitely the graph are more expensive, because you need to revisit different branches in the graph and do the alignment over there. That is more workload than you just have one reference genome that is linear representation. But still, the graph representation is more efficient than comparing with individual reference genome. If you have 10 reference genome, for example, the more efficient way to represent them, just use a graph. In this paper, they said, okay, yeah, it's the time to change the reference genome. Should we have one reference genome per city, per country, per continent, or whatever? And this is, again, what they call pangenomics. Okay, now come to the computational time. When we have the analysis between the sequencing machine and the read mapping step, we see that sequencing can do really good, such that they can read one human genome in 1 hour. But when it comes to the analysis, it's really bottlenecked in read mapping. We need a lot of resources to do this analysis. Again, this depends on what tool you are using, what resources, how fast these CPU hours, and so on. And now let's do the analysis why read mapping is a bottleneck in this pipeline. What makes it a bottleneck? First, one reason is that we have a huge amount of data generated by the sequencing the machine. And this data is going to grow more and more over time because sequencing technology become cheaper and they have more parallelism these days, such that you can handle many samples in the same time, the same sequencing run. The second fundamental reason, we are using specialized machine for sequencing, but general purpose machine for the analysis, which doesn't make sense, right? If you are investing in sequencing and that is a must step after sequencing, then both of them should be specialized such that you know what you are doing. You have a machine that knows exactly what are the needed operation and only perform this operation in a very efficient way. Until now, we are still using the Neumann model from 1945, as my colleague were explaining in the last few lectures about processing in memory, processing using in memory, where the storage is separated from the computation. So you need to move the data from the storage to the main memory to the caches. all the way to the CPU cores to do the processing. And that is what we call data movement. So data analysis is still performed far away from the data. And when we do the analysis for all these kind of data movement, we see that it's really expensive. It's dominate even the performance. So a single memory request consumes at least 160x more than performing computation at the CPU side. So just moving the data over there, that is almost 40% of the total energy spent for the analysis or for the overall system energy. Now, this is a tool called Minimap2, which is do the read mapping. It's a state-of-the-art read mapping tool. And when we do breakdown analysis for the time that it takes to do read mapping for a single read set, we see that more than 60% of the time is done or spent in the last step, that is sequence alignment. So most of the time is spent only in calculating the edit distance. Whether that is a good thing or bad thing, we don't know yet, but I'm going to explain now. Let's see if it is a good or bad thing. Now we are using for the sequence alignment dynamic programming algorithm. Why is that? So think about this example. We want to compare Switzerland and Netherlands as a word. We want to see how many differences over there. In theory, we can compare Netherlands to the S, Netherlands to the S and W, Netherlands to the S and W and I, all the way until we compare with the full word. Now you can see a lot of repeated computation over there. So when you compare with the S, then you compare with the W, then you compare with the S and W, try to figure out where's the good location for S and W. That already include the computation you did over there. Likewise for all these steps. Now for these kind of problems, the best algorithm to be used is dynamic programming table. Why is that? Because you store the previously computed value somewhere, and when you need it in the next cell, you just revisit that value and use it as it is. That's why we are using dynamic programming algorithms for that. However, this is a quadratic time algorithm. It's very expensive. A quadratic time in sequence length. So if the sequence length is 100, and that is something. If it is 1 million long sequence, then that is really very expensive to perform because you need to build million by million table for the sequence alignment. Now in dynamic programming table, you cannot build one cell before the other cell. So if you want to build this cell, you need to build that cell, this cell, and this cell before you compute that cell. So you need 3 cells for each single cell to be computed. And that limits the way you compute these kind of table. So you need to compute the first row, then the second row, then the third row, and so on. Or first column, second column, third column, and so on. Or anti-diagonal. But there is still an order that you need to respect when you calculate this table. And that limits the parallelism when you want to do hardware acceleration. The third challenge is that the entire matrix need to be computed until you figure out the optimal number of computation, which is 5 in this case. And you cannot stop somewhere in the middle, especially if you are looking for a global alignment where you compare the 2 sequences end to end. So these are the challenges with the sequence alignment. There is a paper from MIT. They mathematically prove that you cannot do better than quadratic time for sequence alignment. You cannot, sub-quadratic time is no possible without having heuristics. Of course, with some heuristics, you can do better. But again, you are compromising something. All right. And the other challenge that makes ReadMapper bottleneck is that 98% of the time we are checking the location, the reference genome, we find them dissimilar. You can expect that because we only use pieces of the read and pieces of the reference genome to match them in the indexing step. So we use small pieces, but they could be still different between these pieces. So yeah, the pieces exactly found in the reference genome, but between every 2 pieces, there might be some differences that make them really highly dissimilar, and thus they are not interesting for genetic variation discovery, for example, because there are too many differences, and that lead that probably this location is not a good fit. All right, so to solve all these bottlenecks, all these problems, you have the computer stack. You can start from the tops where you can improve the data, maybe. You can compress it, make it less in size so that the computation maybe can be faster. You can change the algorithm, for example, make it faster. Or you can play also with the low level, such that you build specialized hardware to do the processing. You change the logic for the computation from CPU to FPGA to GPU to ASIC, maybe build customized chip for that. If you want to start with the software level, here's an example of matrix multiplication where you have different implementation. You can see the running time. So if you use Python, it's really something bad. You don't want it, especially when you are looking for high performance computing. If you use C, you will see about 50x of improvement. But if you start using hardware or SIMD units for the computation, you start to gain a lot. And this is exactly what we can see from genome analysis. It depends on what kind of optimization you are looking for. This is a real experiment done by Hingley. I think the link is hidden over there. Yes, this is the GitHub link over there down. You can access it and see the code for that. You can play with it yourself. That is to read the FASTQ file, which is the read set, the file where you have the read, just to read the file. That is IO cost. If you use C, see the running time. If you use Python, then the runtime really shoot up by maybe 9x, and here by 4x. That is depends if it's compressed data or plain data. So you can get more information about the importance of efficient programming plus efficient hardware for that. And that, to sum up, we really need intelligent algorithm and intelligent architecture that handle data well. Not one of them, but both of them really necessary. Okay, now let's start with maybe the most interesting part, or maybe the most boring part. I don't know, it's up to you, which are our own work from Safari Research Group at ETH Zurich to improve all this story. So this is the first paper where we list or overview all existing methods, try to improve every single step in read mapping. So read mapping is a 3-step process where we have indexing first, we build a hash table, then we do what we call pre-alignment filtering. That is to check the quality of the read and the reference genome before do sequence alignment, before do dynamic programming. So we check the quality using some heuristics that tell you quickly if it is good fit for that or just ignore to save some computation. And also the other algorithms that try to improve or optimize sequence alignment. As I told you, you cannot do better than quadratic time, but sometimes you can use heuristics, depends on the application. And there are many heuristics over there that we list them for accelerating sequence alignment. If you're interested in working on GPU FPGA acceleration, this might be a good reference for you. Okay, so now we are going to list 3 main directions. First one, seed filtering, which is related to improving indexing. The next direction, what we call pre-alignment filtering. As you can infer from the name, it's a pre-step, step coming before the alignment step. And also the third step, which is read alignment itself or sequence alignment. All right, and here the summary of the hardware acceleration contribution of our group. So we have the sequencing machine, the storage, the main memory, and the CPU. And first, we try to improve the CPU performance by replacing it by FPGA or GPUs. We have several works in this field. We also try to do the computation inside the memory or near the memory. And we have a number of papers on that. We also try to do near memory sequence alignment. And we have a paper over there. We have still ongoing projects with other part of the systems inside here, inside, and these hopefully to be published soon. Then we can explain them in the next semester, hopefully. Okay, let's start with the first one. This paper called Fast Hash. It's coming from Onur Mutlu and Can Alakan, my PhD advisor. And they proposed 2 mechanisms to improve hashing or seeding. the way we collect seeds from the reference genome or the read. The first one called adjacent k-mers, and I have figure to explain. This is the read, this is the reference genome. We want to collect seed from here, seed from there. Assume we collect the seeds from the read that they are adjacent to each other. For example, this is one seed, all red. Here, probably it's yellow, but here it's brown, maybe. Yeah, but this is another seed, and this is the third seed. Now we want to look up in the hash table, we find location here and there and there. And now the things I was explaining before, it was not used until this paper proposing adjacent k-mer, which is the same concept that I was explaining. When you are looking for this seed, it should always be before that seed, and this should be before that seed. This is how we call it adjacent k-mer. What does that mean? When you look at all these locations, it seems this is the most optimal location where the red come before the yellow, before the green, and they are adjacent to each other as we have in the read. So this is probably the most similar location to the read. Here you can see only the red, but the yellow really far, which can be deletion, or maybe it can be wrong location. However, we are dealing with short reads here, so we don't care about long or large deletion as we have here. So this paper, I think, was proposed in 2008 or so. Let me see, or 2013, I think, fast hash. Yes, 2013 over there. So in that time, we didn't have long reads. Yeah, so this is the key idea of adjacent k-mers. If they are adjacent in the read, then they should be adjacent in the reference genome. And that improves the overall read mapping time a lot. The second proposed mechanism, which they call cheap k-mers. What we mean by cheap k-mers? So there are some k-mers very expensive to process, and there are some k-mers very cheap to process. I have an example also for that, better than the text. Think about this read sequence, and we have a frequency threshold of 500. What does that mean? It means when I'm looking for a seed in the reference genome, it should be less than 500 times appearing in the reference genome. If it appears more than 500 times in the reference genome, then I consider it as expensive to be examined, and thus I ignore it. I don't check it. Yes?
Is the k-mer the same thing as a seed?
Yes, yes. I think I mentioned that before, right? The k is the length of the seed. So you can replace k with 9, for example, then it will be 9-mer. That is the same as seed, or subsequence coming from the read or the reference. Yeah. So the frequency threshold is 500, means I'm looking for the seed that appearing in the reference genome less than or equal 500 times. What does that mean? So I'm checking one of the seeds. Assume it appears in the reference genome 4 times. These are the location. Let me maybe remove this highlight. Yeah, these are the location of that seed, and this seed appears only 4 times. I'm checking also other seeds, and I see here it appears 2 times. Here it appears maybe 1,000 times. Another seed appears in the hash table 2 times. Other seed appear 2,000 times, and so on. Based on the cheap k-mer definition, I'm going to examine only the k-mers that appear less than the threshold, which is only this list, and that list. And I'm going to ignore all these lists. Why is that? Because these are probably not interesting for me, because they lead to too many similar locations in the reference genome, and I need to examine all of them. There is nothing distinguishable that I can rely on so that I can say this read is only coming from this region, not from all these regions. So this I can consider it as a noise. For that, I will use some of the list, so you can compare before and after. Sorry. Yeah, this is the location that I need to examine before, and this is after defining cheap k-mers. I only have 8 locations to be examined, and that is a huge reduction in the workload for testing these seeds. Yes?
Is it possible to use run length encoding to discard some of these locations?
Run length encoding. Yes, if they are similar, because run length encoding is about a consecutive character that they are similar. So you have like 100 Cs. and you want to represent it as just 100C. But here, it's about the frequency of the seed. The seed might not be the same, like all of them Cs. The content might be just heterogeneous, right? All A's and C's and G's and T's, right? So it can be anything. We don't need to restrict it here, but just the frequency of that seed might appear in many locations in the reference genome. Now, yeah, run length encoding is one way to compress reference genome, for example. We use that for compression. It might not be efficient, yes, but there might be other more efficient algorithm. If you are asking about, we merge the run length encoding for compression and this seed filtering as one algorithm, that could be something interesting. I'm not aware of any algorithm doing that, but could be something interesting as a fast implementation of this idea.
Right, because that can run length encoding, basically run length encoding or some sort of, what I'm imagining is a frequency counter which counts the number of occurrences of each character in the read, the seed. And this frequency can be compared very quickly as compared to a full read mapping. Yeah, maybe we can discuss that.
There is a bit similar idea called weight minimizer. I think it was published in ICMB or Recomp 2 years ago. Very recent one. I can point you offline, probably, but they call it weight minimizer. I think it's from Adam Flippy group, where they try to rely on the frequency to reduce the size of the hash table. So instead of storing all these seeds in the hash table and then check their frequency, they check the frequency before they store it in the hash table, and if it's exceeding that threshold, then they ignore it from the hash table. They don't store it at all in the hash table. In this way, they have a small size index that is very quick to query. That is something implemented, but run length encoding might be interesting. Yeah, thanks. Yes.
Just to make sure I understand it correctly, do you keep this? So in total, do you keep the same number of k-mers, or does this also reduce the number of k-mers used for the alignment?
When they propose this, the number of seeds in the hash table is the same. You don't change it. You have one index. You keep checking the frequency. You decide to take, not take, based on the frequency. But as I said, the proposed method 2 years back, I think in Recomp, if I'm not mistaken, or it could be Recomp-seq. I can send you on Moodle the exact paper. It's called weight minimizer. In that work, they change the index. They make a modified version of the hash table, and they use that always, and they ignore the original index or the hash table. Yeah, thanks. So you can see the things there that you can do at only algorithm level. So all these changes at algorithm level. That is the conclusion. I'm going to skip just to give some time for the other works. All right, so we finished from seed filtering. There are many more works, by the way, from other research groups, but I'm trying to focus on the things that we have done recently. If you're interested, read the paper I mentioned earlier about all existing work from other research groups. Now about pre-alignment filtering, which is, we know that now, hopefully, all of us agree that sequence alignment is expensive, which is dynamic programming algorithm. And our goal is to reduce the need for dynamic programming algorithm. We cannot get rid of this dynamic programming algorithm. As I said, it's the only algorithm that can give you the optimal number of differences between two sequences. So it's something you cannot avoid. But you can reduce the input workload. If you know these two sequences are really different by 10 differences and your threshold is 5, then probably this is something to ignore. You don't need to compute dynamic programming to know that these have 10 differences, for example. And I'm going to show you an example. So genomic strings, they are two categories, similar or dissimilar. The dissimilar are expensive because you waste computation on them, and at the end you ignore them. Simply, you don't need them. So you use dynamic programming here, but at the end you just delete them, flush them out. And you need to ignore them if the number of differences exceed the threshold. So this is the definition of how you define it, dissimilar or similar. But if they are similar to each other within a threshold, then it's interesting to find the number and the location of all these differences. And this is how we call the pre-alignment filtering concept. So you have the sequences going to the pre-alignment filter first, and then if they are good, if they are similar to each other, they go to read alignment step. However, you need to maintain three things if you want to design a pre-alignment algorithm. First, you need to filter out most of incorrect mappings or dissimilar sequences. You cannot filter all of them, otherwise you will be optimal algorithm, and that should replace sequence alignment, so that you cannot be optimal. And for that, we said most of them here. Second, you need to preserve all similar sequences. You don't want to compromise accuracy again. This is a very important point. Third, you need to do it quickly. You cannot be just as taking the same execution time as read alignment, for example. You really need to be very quick so that you can decide quickly here, filter most of the bad things, and keep only the good sequences for sequence alignment. This is the first work, Gatekeeper, we proposed in 2017. And the key observation here is that if two strings differ by E edits, then every character can be aligned in at most 2 E shifts. The key idea is to compute shifted Hamming distance, which is AND of 2 E plus 1 Hamming vectors of 2 strings. I have an example to explain that. And then we use pit parallel operation to nicely map the architecture we built to the FPGA. The results were really significant compared to existing CPU version. And here's the example to explain that. Think about Istanbul and Istanbul as 2 words to be compared together, okay? Now, if you do Hamming distance, exactly match, no differences, right? Now, assume myself, I want to impose a deletion. I want to delete 1 character from there. What will happen? I will delete the A, then this version will be 1 word, so I need to shift these here. Now I have a new word with a deleted A and the original word, and I now want to compare with these 2 words. If I do Hamming distance again, I will get 5 mismatches, although what I did, just 1 deletion, right? So apparently XOR between the 2 words does not work here. What do I need to do? If you notice that when I delete 1 character, all these folks over there shifted to the left, right? So I said, why not shifting the entire word here to the right and then do the comparison? What does that mean? See this slide. Now I have this without the A. Now I will have another copy that is shifted to the right, still without the A. So this is a version of only this guy, but not that guy. Now when I do the comparison, Hamming distance between this and this, and this and this, you will see that we have 3 matches over there and 4 matches with the second guy. Now, because I use XOR, this is the result you get from the XOR. And when I do AND operation between these 2 vectors, this is what I will get. What you can see here, you can see whenever it match 0, mismatch 1, and also the 1 represent the thing that I deleted myself. So I deleted the A, and the A was located here. So this pattern tell you that there was alteration over there, which is, you can see, directly correspond to the A, and everything else remained the same. So this is the right way to do the comparison. This is what we propose in the paper. So to do shifted Hamming distance, but not Hamming distance. So whenever you have a variation, do multiple copies of one of the sequences where you shift it to the right and left. You need both of them because you don't know if the original variation is deletion or insertion. It could be both of them as well. Could be an insertion and deletion all together in the same sequence. That's why you have 2E plus 1 copies of one of the sequences. What does that mean? Let's see a real example. There's a real genomic sequence. This query and this reference, we want to compare them together quickly without using dynamic programming algorithm. So we said generate 2 E plus 1 sequences. What is E? E is edit distance threshold. So if your edit distance threshold 3, then you are going to generate 7 vectors as we have here. So we have 7 vectors because I set myself the E to 3. What does that, how we generate all of these? The first one is just XOR, just Hamming mask. XOR between these two, you got all these. This group, I call it deletion mask, is where I shift the query by 1 step to the right and then compare with the reference. The 2 deletion mask, I shift the query to right again, 2 steps, not 1 step, 2 steps, then compare XOR with the reference. 3 deletion, compare with 3 steps or shift the query by 3 steps to the right and then do XOR with the reference genome. For insertion, I do the opposite. I do the shifting in the opposite direction, which is left. Yeah, this left, not this. So I shift to the left by 1 step and then do the XOR operation. And then I do the shift by 2 steps, then do comparison and so on until I get all these factors. Now, why I consider shifting to the right over here and shifting to the left over there and no shift over there? Hopefully, this way I can cover all type of variation within edit distance threshold 3. So any type you can think about, 3 deletion, 2 deletion, 1 insertion, 3 insertion, 3 substitution. All of them I can cover using only these 7 vectors. And you can give me an example, I can show you how I can capture these variations. Let's see the example here. Since I do XOR here and I got zeros, it means this portion of sequences is exactly the same. And you can see it from the 2 sequences, actually. They are really the same over here. That's why I get a bunch of zeros. And then I got 1, it means there is a T, but here there's a G. So there is substitution over there. And then I get a bunch of zeros, and then no more a bunch of zeros. You can see it's just random. However, I got a bunch of zeros in the second vector. What does that mean? It means a deletion happening over there. You can see there is 1 character deleted from the C. Here there are 2 C's, but here 1 C. So there is deleted C over there that cause the matches to appear here, but not over there. That's why you can start seeing the zeros over there appearing on what we call 1 deletion mask. So if you get a lot of consecutive matches on that mask, it means there were a deletion before. Likewise, if you got a bunch of zeros over here, then there is an insertion before these zeros appear on that mask. Let me highlight them, might make it easier for you. So these zeros, they are the largest number of zeros in any given vector. You can see, you cannot have larger than these zeros in this area, for example. Same thing here. You cannot have larger than or longer than this 0 chunk over here. Same thing with this. This is the largest in the entire area over here. And this is the largest in the entire area over here. So we leverage this, these pet vectors, this algorithm, to calculate quickly how many differences are there. And then we use AND operation to give me this vector, and then I count the number of ones. There's 1, 2, it means there are 2 differences between these 2 sequences. If they exceed the threshold, it means bad sequences, so ignore them, don't perform sequence alignment. Yes?
Can you determine the number of shifts? Does the number of shifts will determine the runtime of the algorithm?
That's true. So again, as I said, we need to build 2 E plus 1 masks. So E is the edit distance threshold. So it's up to you to set that threshold. You can say, I'm not interested in any 2 sequences that we have more than 100 differences or more than 1 difference. That's up to you. Of course, setting that parameters will directly impact the runtime because you can see the workload here is exponentially correlated with the number with the edit distance threshold. So every increase in 1, you cause at least here 2 increase or at least 2 increase in the number of vector or 2x probably. Yeah. Any questions? Go ahead.
So just on a high level, the motivation for this is that it's like maybe like it's really good, but it's not quite as good as the full dynamic programming system. But using this, you can probably rule out like 95% or something of couplings as being dissimilar strings. Right?
Excellent. Yes, exactly. This is the goal. So the goal is not to find where are these differences or how many of them exactly. The goal is just to tell you whether the differences exceed or they don't exceed the threshold. I don't care if the threshold is 10 and I'm telling you you have 100 differences. Even if the number of differences is only 12, for example, I don't care. As long as it exceeds the threshold, I don't care about the actual value, if it is 100 or 50 or 60. As long as it exceeds the threshold, then done. These two sequences are dissimilar by definition. Likewise, if they don't exceed the threshold, if my algorithm said these two sequences has 2 differences, and in fact they have 4, as long as both values still below the threshold, that's fine. But if they start to contradict each other, there is a problem. If the 2 sequences has only 1 difference, and my algorithm is saying they have 100 differences, then that is false negative. I don't want these cases. So I don't care about the exact number of differences. I don't care about the location of the differences or their type. I'm not interested to know if they are insertion, deletion, or substitution. In this stage, only I would like to filter the sequences that they have high number of differences, exceeding the threshold. That's it. So I want something very simple, very quick, that can do this operation just to reduce the number of times that I'm going to use dynamic programming algorithm. As I said, I cannot avoid using dynamic programming, but I can reduce the frequency of using that expensive component.
Okay, so under the added threshold, this does not have enough accuracy to choose the best coupling.
Yeah. As long as the number is below the threshold, I don't care. I don't want to impose any accuracy on improving that time. But as I said, both of them should agree. If the actual number is less than the threshold, my algorithm should always say it's less than the threshold. Likewise, if it is exceeding the threshold. I don't want any false negative, but it's okay to have false positive. Meaning that if the 2 sequences have more than the threshold, and my algorithm is saying, no, they don't exceed the threshold, they only have 0 differences or 1 difference. That is false positive. It's okay to have it. Why? Because we have more rigorous step after that, that is sequence alignment. That is dynamic programming algorithm. It's going to rigorously check everything and filter them out directly. But we don't, again, want to have a lot of false positive. Otherwise, no benefits of having our own added step. Okay, hopefully I can cover much of the material by now. There is a problem with the approach that I presented over there because I'm using AND operation to AND all these path vectors. Sometimes there is differences here. There are 2 ones, part of the correct solution, but they are not captured over there. You can see there are zeros. Why? As you know, AND operation, whenever you have a 0, it will always dominate anything else. If you have 0 and 1, directly you will have a 0. To just try to reduce the effect of this or using AND operation, we did a hack, replace every single 0 or 2 zeros by 1s. That is a heuristic. Replace every single 0 or 2 zeros with 1s. You can see here, the result is less zeros, less number of zeros over there. And here we have single 0, no longer single 0. 2 zeros over there, no longer 2 zeros, but more than 2 zeros, just keep as is. What does that mean? We know that any 2 sequences, when you compare them and they have a low number of differences, then there are going to be subsequences that are always large. Think about the green highlighted segments over here. These pieces are always long. 1, 2, 3, 4, right? So it won't be harmful to replace the pieces that has short zeros, like 1 or 2 zeros, because anyway, most of the time they are not part of the solution. So we are not going to change the actual sequence. It's not changed here. You can see we didn't touch these green pieces. They are still the same. What we are trying to do by replacing these zeros is just to have a proxy for these errors so that they appear in the final result. So we try to ignore all zeros in the surrounding area so that when we do AND operation, the ones will appear over there. So now when we do the AND operation, we try to fix one of the issues, but still one of them is missing from here because one of them appears with large chunk of zeros rather than 2 or 1 0. So we cannot solve that. Now the question, how we choose 1 or 2, or can we choose 5 0 to be replaced? Can we choose 6 zeros to be replaced? Yes, definitely. You can do that, but we didn't test this in the paper.
Destroy information like negation.
Yeah, as I said, the zeros always mean match. We don't touch the ones, we touch the zeros. The ones still there, and we try to keep them. If there is one in the correct solution, as in have here, we didn't touch it, but we touched the other noisy zeros over there. So if there is a 1, then probably this one is coming not from the zeros that were replaced or changed. It's probably coming from the mutation itself. The observation we made is just that whenever we have similar sequences, they always have these long segments of zeros. That's why we always go to the short ones with zeros and tweak them. We can play with these because they are noise, basically. So we try to solve this problem and apparently try to improve the algorithm. And the good thing with this kind of algorithm, this dynamic programming algorithm, but this is Gatekeeper. And you can see in Gatekeeper, we try to shift the entire thing and compare it with that has no dependencies at all with any cell. So you can compute this cell before this, you can compute this before this, you can compute that before this, and so on. You can do whatever you want. All pit parallel operation. Again, I'm going to show this in case you forgot it. So we shift the entire thing, we compare it with that. So you can shift this individually, compare it with that. Shift this character individually, compare it with that. So that you can build the entire vectors over there. So everything is independent of each other. You can build this bit over there, apart from that. You can build this apart from that, and so on. This is something we are missing in the dynamic programming. Dynamic programming, as you can see in the direction of these arrows, this cell required this and this and that to be computed in advance. Here we have high parallelism. You can build the entire thing in one shot. You don't need to wait for anything. Just use the FPGA logic to shift the entire thing, build the entire thing for all vectors in the same time. You don't need to build the first vector, then the second, then the third, then the fourth. And for that, you can nicely map it to most of the hardware accelerators, such as SIMD, FPGA, GPU, and so on. However, we choose FPGA because we see most of the operation rely on AND, XOR, shift, which are FPGA optimally designed for these. Now, these are details about the FPGA implementation. This is one important thing when you're targeting hardware. Normally, when you target hardware, you need to know what you are targeting. You need to know the architecture of the FPGA that you are dealing with. Here we choose to have 5-input lookup table. Why is that? Because when we want to replace 2 zeros, we need to check 2 bits to the left, 2 bits to the right to know that if the pattern is really 1001 or something else. Because if there's 3 zeros, we really need to check 2, 2. But if it is 101, it's easy to check. We don't need 5 bits. So these are the 2 cases why we need 5-bit or 5-input lookup table. If you still suspect that or you don't believe it, we can discuss it offline. But this is how we need 5-input lookup table. And we choose an FPGA that has at least 5-input lookup table so that we do optimal implementation where we need only 1 lookup table per bit. Otherwise, if we use 4 input lookup table, then we might need 2 lookup table per bit. So what I want to highlight here, it's really important to know what you are dealing with when you use the architecture so that you can optimally utilize all the resources. This is how we utilize the FPGA chip, and these are the results we gain. Apparently, you can see speed up from that. I'm going to skip the next one, which is Shuji, but the video is already online. And I'm going to explain Sneaky Snake quickly, which is one of the latest work we did. Yeah, so hopefully you can still see the slides. Okay, great. So in Sneaky Snake, or in this paper, which is our recent work called Sneaky Snake, that is a pre-alignment filter work for CPU, GPUs, and FPGAs. And the thing we observe over here, if you have a sequence, another sequence, when you compare with it bit by bit, like when it match, you add a dot. When it doesn't match, you add a space, nothing. You see the matches are always around the diagonal, and they are always long. So this is long segment of red or blue to represent the matches between the 2 sequences. We have a sequence here, sequence there, and these are represent the good solution where it tell you where are the matches and non-match and mismatches. So as observation, we see that correct alignment is a sequence of non-overlapping long matches. And our goal is to collect these non-overlapping long matches. So in this work, we try to map the problem to VLSI chip routing problem. If you don't know, this is the VLSI chip. If you dissect the chip, you will see something like this, where it has component. And our goal is to move from this point to that point with the least routing cost. So we don't want to pass through many components in the chip, because every time you pass, you need to detour or go through the chip, and you need to a port here, a port there, and so on. So every component passing through it will be very expensive. Yes. Oops. Yeah, maybe. Thank you so much for highlighting that. Let me end the show. Share the screen again. Yeah, feel free to leave if you have a class after this. Okay, let me share it. How's that? Yeah, I think what they are seeing. Okay, now they see the slideshow, not the presenter mode. Okay, yes. So this is one of the work we did. We try to avoid all these obstacles on the way while we are doing routing. As I said, it's very expensive to avoid obstacle. We consider this system on chip with many components, each component as obstacle. And our goal is to build pre-alignment filtering, leveraging these routing algorithms. So what we did, we have an equation to build this table. So you can build it using this equation. It's different from Gatekeeper. But the common thing between them that we use edit distance threshold, and the number of rows in this matrix is similar to Gatekeeper, which is 2e plus 1. So number of rows here, 2e plus 1, which is 7. However, when we have this matrix out of the equation, we convert each one as obstacle, each 0 as non-obstacle path. So we convert this into this. Again, this into this. And now the problem becomes passing from here to there while passing through the minimum number of obstacles, which are the black boxes. So one way to solve it, remember, you have a limit of 3 components to bypass. How we solve it, we go row by row, we check how many steps we can go further straightforward. So we see that this row allows us to go 4 steps forward. We choose that path because agreedly, this path always send you to the the optimal path where you have low number of obstacles. The shortest the path, the fastest, or the path with less number of obstacles. Otherwise, if it is not shortest, then you are definitely detouring to something else that makes the path going longer and longer. So if it is going straight forward from here to there, if you find such path, that always will be the most optimal one with no black boxes in this. That's why we greedily choose the one with largest number of white boxes. So once we face an obstacle, we need to reduce that from the limit over there. Now the limit will become true. Now 2 obstacles we can avoid. We repeat the same steps. We check every row, how many steps we can move forward. We see that this path provides the longest path or the shortest path overall, because there is no detour to there, for example. And we choose that path. We see an obstacle over there. We reduce the total number by 1, and we repeat. Now, if all of them has the same amount of obstacles or same number of steps that you can move forward, doesn't matter which one you choose. You can choose this or this or this, because all of them have the same penalty, which is reducing this by 1. So we choose this, we reduce this by 1. Once it's 0, we terminate the algorithm, regardless we reach the exit or not. If we are in the middle here and we reach the 0, we terminate directly, and we say there are a large number of obstacles that we cannot avoid anymore. Then these 2 sequences really different. Just ignore them. This is in reality what we built, not the previous one. Why is that? Because when we check the row, we see an obstacle, then we need to terminate over here. So you don't need to continue calculating. When you see not obstacle, you keep continuing until you face an obstacle, then you stop. You move to the next and so on. That's why even for CPUs, it's still really quick. It's a very quick algorithm. And when you implement it on FPGA, the resources over there, it's compatible to what we have in Gatekeeper. So you can see these resources for Sneaky Snake, which we call it Snake on Chip if it is FPGA. And this is the Gatekeeper. The number of lookup table, it's still very low for one sequence. This is for one problem. Single filtering on it. These are the results. I'm going to skip. You can see significant improvement. Again, we still have data movement over there because we are using FPGA GPU. We need to move the data from the main memory all the way to this hardware acceleration. So we try to improve this by doing processing in memory or processing near memory. I'm not going to introduce this because these are the previous lectures. I assume all of you are aware about the topics of improving the processing near memory. But what I'm going to explain how we improve Sneaky Snake to be able to do the processing near the memory without the need to move the data far away from where they reside. So this is the hardware we used. We have Power9 from IBM. We use CAPI2 and OpenCAPI as 2 alternative connections to move the data from the Power9 to these FPGA boards. This FPGA has HPMEM memory, where it's integrated, tightly coupled with the FPGA chip on the same package. They are not separated from each other. So the memory and the FPGA, they are on the same package. So they have very short interconnects between them to reduce data movement. This is normal FPGA, where you can see it in almost all FPGAs, where you have DDR4 or DDR3 connected with the FPGA with long interconnects. Now, once we do this with careful implementation on this FPGA, you can see the improvement. The blue one over there, this line is the DDR4, and the orange one, where we use multiple ports from the HPMEM cube. Apparently, you can improve even for single processing element. That means single problem or 2 sequences processed in the same time. But here, 4 sequences processed on the same time because 2 problems. Each problem needs 2 sequences to be processed. Here we need 3 components, that is 6 sequences and so on. When we use even processing just 2 sequences in parallel or in the same time, you can see HPMEM provide improvement over DDR4 because we have high bandwidth and shorter interconnects to move the data over there. That is with 4 ports connected to the HPMEM cube. But when we use 1 port, the red one over there, you can see HPMEM is not better than DDR4. Why is that? Because transfer the data as 256 bits on one chunk, but the DDR4 transfer 512 bits per chunk. So you can see that you can move more data in DDR4 compared to HPMEM single port. But when you use multiple port, you cut down the processing time over there. As you are using processing near memory, as you expect, you have a lot of energy saving because of that compared to Power 9 with 64 threads, 16 cores. So these are very significant improvements just because we changed the hardware to another hardware plus some careful changes in the architecture we have for Sneaky Snake. That is Sneaky Snake, more details about it.
Mohammed, do you hear me?
Yes.
So we can't actually see your screen in Zoom and also in...
Oops, I think the GoPro is disconnected again. Yeah, let me see the laptop camera. Do well. Yeah, thanks for the heads up, John. Sorry for that. I don't know why the GoPro keep disconnected.
But it's actually the screen share that's the problem, I think.
Yeah, I agree with Joe.
What do you mean? Should I stop sharing?
Maybe try to share again, yeah.
But we can see it as shared, right? I don't see any problem with it. Let me share again. Can you see it now?
Yeah, now, yeah.
That's a bit weird. I don't know. Thanks for that. Yeah. So with this work, it's a Grimm filter, another filter for processing in memory. I'm going to skip that and jump to the processing near memory for sequence alignment. So this is the last work that I'm going to present for today, and it's covering read alignment or sequence alignment processing. Yeah, please let me know if you don't see the shared slides. I'm moving the slide now. I'm slide 211. And this is the work that we did in our group called GeneAsm. Try to do many things more than just sequence alignment. So our goal in this work is to accelerate approximate string matching problem, which is sequence alignment, and design a fast and flexible framework. We call it framework because try to do much more than just sequence alignment. And now I'm going to tell you what it does. So the key ideas of this work is first to exploit the high bandwidth memories as we did in Sneaky Snake and the logic layer of the cube. So you are not using FPGA tightly coupled with the HPMEM memory, but rather you are using the logic layer inside the HPMEM cube. And the second thing, we leverage a very old algorithm called BiTAP. It was proposed in 1992. However, that algorithm was doing great, but missing something very important, which is supporting long reads and traceback. Traceback is the step that we use after we calculate the entire dynamic programming table and tells us where are these differences and what type are they. This is what we call traceback. So this BiTAP cannot do this. And also it's not highly parallelizable. So we are trying to solve all these three issues in this design and then co-design the modified version of the algorithm along with the processing inside memory. So the results were very significant for three use cases: read alignment, pre-alignment filtering, and edit distance calculation. Even compared with Darwin and GenX, which are, GenX, for example, is ASIC design. Darwin is ASIC and FPGA, and you can see improvement even compared with those. These are CPU tools. This is Shuji pre-alignment filter on FPGA that I was supposed to explain, but because of the time, we just skipped it. But I'm going to send you pointers after the lecture about the video that we explain about all this work. So if you're interested, you can watch them. This is edit distance calculation, does not require, for example, trace back step, because this only tell you the total number of edits, which is edit distance. So you don't need traceback in this case. So you can see the speed up is much more than what we get in sequence alignment, because the computation here are less and the hardware is less complex than what we have with traceback. This is the summary of what we did in the past, all of this work. So I didn't give a just to all these. I need a really separate lecture just to explain every and each of these work. But if you're interested, I'm happy to send you a pointer, as I said. And as a conclusion in this lecture, read alignment can be substantially accelerated using computationally inexpensive and accurate pre-alignment filtering algorithms designed for specialized hardware. Again, I cannot emphasize more. You always need a new algorithm and new hardware architecture, not one of them. So better algorithm is always good. Better hardware is always good, but designing algorithm that is aware about the hardware, what you have, what you can leverage, what you can exploit, that will give you the best benefits out of the entire both field. All three directions, seed filtering, pre-alignment filtering, sequence alignment, are used by mappers today, but filtering has replaced alignment as the bottleneck. As you can see, all workload go to pre-alignment filtering first, be processed over there, and then only a few of them will go to the sequence alignment. So we push the workload from sequence alignment to the pre-alignment filtering, and that is what motivated us to focus on pre-alignment filtering. We have a lot of work on pre-alignment filtering. Pre-alignment filtering, the good thing with it, that does not sacrifice any of the aligner capabilities. Since we do the heuristic or the computation at the pre-alignment filtering stage, and everything that is good, it just moved to sequence alignment. So whatever feature you have in sequence alignment, it's still there. That is what makes our work superior to the existing work when you try to accelerate Smith-Waterman, for example, or Needleman-Wunsch. Then you need to do some compromises over there such that you gain fast performance over accelerating these. What else can be done? Think about this again. What if a new updated reference genome given to you? Do you need to repeat the entire read mapping step? This is our next work, which we call airlift, where we don't need to repeat read mapping if you got a new reference genome. Assume you already did read mapping before. Now I'm giving you a new copy of the reference genome. I told you that it was updated in this chromosome. We solved this gap. We improved the accuracy over here. Then we need to leverage this information where the new reference genome is not totally new, but rather some portion of it updated. Then we can leverage these to prevent performing read mapping again, which is very expensive. So that was the key idea of airlift, where you can detect what has been updated, what has been remained the same, and leverage this information to improve read mapping performance. That was all for all hardware acceleration work. We have much more than this as ongoing or not being explained today. But what I want to answer, the question that I asked you in the beginning about whether sequencing technologies are going to get better and read the full sequence or not. But before that, the last important step, and many of the people ask us when we present all of this work in journal or in conferences, we don't see much of this hardware accelerated work adopted in real genome analysis. Why is that? We got this question actually in December 2016 when we submit our first work as a gatekeeper. Let me try to remove these. Yeah, this is what we got from the reviewer. I copy-paste his reply. So I have a major concern with the work that is actually not a problem with the manuscript. There has been little to no adoption of previous specialized hardware solution. Why is that? And I attached our answer, copy-paste again from our response, it's 2 slides. But in summary, we said it always takes time to adopt a new or different hardware technology. If the entire community was using just CPU algorithm for genome analysis, of course, it will take a lot of time to build infrastructure for these to use FPGA or GPU in a hospital or in clinics. And we were right. That was in 2016. And we were dreamer at that time. Nobody was using FPGA for pre-alignment filtering or for steps of genome analysis. And after 2 years, a company started to adopt these FPGA devices to be inside the sequencing machine. This is a real device called Dragon, which is FPGA board inside Illumina devices. This is another example from NVIDIA. 4 years later, they start to develop GPU cards for genome analysis only. They are just dedicated for genome analysis. And you can see the cut down in time. So computing is still bottlenecked by data movement, even with the commercialized solutions from Illumina and from Dragon, because they are still FPGA and GPU, but not processing in memory or processing in storage or processing in sequencing machine. So we still cannot answer our own question about where's the best place to do processing for this huge amount of data or the genomic data. However, these are challenges to encourage everyone to adopt the hardware solution if they are still not encouraged yet by the entire slides or the entire lecture I presented. So first, you need to accelerate the entire read mapping step. That is a challenge to encourage everyone to use the hardware acceleration. Because if you provide a solution only for pre-alignment filtering, they said, okay, when I integrate it with a full system, then based on AMDAL low, the acceleration ratio will be limited. If you provide 100x speed up, when I integrate it with a pipeline, it might be just 2x of speed up, or maybe less. That is a challenge. Reduce the high amount of data movement. If you provide FPGA chip for that or GPU, you are not solving the issue with the data movement. And you need to really find a solution such as working with the data or the algorithm to compress the data such that you can move the data on higher throughput. You need to develop flexible hardware architecture because users who are a medical doctor or biologist or bioinformatician like to play with the parameter and see the effect on the accuracy of the interpretation. In hardware, we always like to fix the range for each parameter. For example, that is the threshold. Okay, I'm going to set it from 0 to 5 because I'm going to need this much of logic. So normally, this is a trade-off between flexibility and the efficient use of resources. We need to change the data format. As you saw, we are wasting a lot of resources in FASTQ file. We are saving the ACGT data as a text format that is readable by human, which is representing each character by 1 byte or 2 bytes. That is too much. We only need 2 bits to represent ACGT data. And we don't need to move all this huge FASTQ file to the FPGA. We can encode it into 2 bits, and then we are done. Again, accuracy is something we don't want to compromise. The high hardware cost, of course, you are going to invest for more FPGA, more GPUs. That will cost you a lot in the first round, but later on, over the long run, you will see a lot of benefits. The long development life cycle, of course, FPGA is not as easy as programming in C, so you need to be ready for that. All these challenges, I think, are okay. They are not that serious, preventing you from using hardware acceleration, especially when we talk about the benefits reaped or the things that you can gain out for public health or for very important problem when we talk about hospital health and so on. Yes.
In what way does using hardware decrease the accuracy compared to software? Does this mean that if you were to implement the sneaky snake in hardware compared to in software, it would be less accurate in hardware?
I didn't mean that. What I mean, if you are looking for something fast, sometimes you are compromising the accuracy. As a lot of work done in this area, you can see the accuracy is always compromised. Not only the accuracy, but some features sometimes are compromised. For example, traceback. Sometimes people try to avoid traceback and they do it at the CPU side because it's hard to maintain the entire dynamic programming table inside the main memory of the GPU, for example, where you need to revisit it again. So they delay these things or try to not do it, for example. There are many other reasons or examples I can give you about heuristics are proposed for hardware acceleration, and these heuristics reduce the accuracy of the pipeline. I think we list most of these in our paper, the one I show you where we have 3 steps over here, if you remember it. where I list all the heuristics, the algorithm, the hardware acceleration. I think I will show it toward the end as well. Hopefully I will keep it in mind and I will point it out to you. But we list all these heuristics that compromise the accuracy in the paper. Did we achieve our goal yet? I don't think so. We still don't have any hardware that you touch it, can read your DNA and analyze it. This question, although all this work that we have done in hardware acceleration, we still cannot answer that question with one answer. There are many ways to push forward, but what we know that we really need customized hardware to do the analysis. Without that, we are not going anywhere. We will still use general purpose computer. So we still need to find a place where we can process this huge amount of data. But me, myself, I see the best place to be inside the sequencing machine because I don't need to move the huge amount of data somewhere else to another party to do the analysis. You can filter most of the data inside the sequencing machine, then just keep the things that you feel is good, keep it for somewhere else. You can analyze it in another machine or something else. But I still believe you can do much more than what we have inside the sequencing machine. Remember, these sequencing machines, they have GPUs, they have FPGA inside them. They have also storage unit, SSDs with large capacity. All of these are leveraged for sequencing, not analysis. So they already have hardware capabilities inside them just for sequencing. So you can leverage all these hardware without paying extra cost because the things already exist over there, and you can do the processing inside the sequencing machine. We have examples from AI. We have really customized chip where companies invest in this. We have another example from Tesla where they have their own hardware. So why not having it for genomics? Yes, we do have from Dragon, we do have from NVIDIA, the examples I showed, but we're still very far from where are going. Now, finally, I will answer the question and then we end up the lecture. What if we will have a sequencing machine that can read your entire DNA in one piece? They don't give you reads, but give you just your full DNA. What will happen to all the things I was presenting? Think about that. Will they just trash them? Will we reuse some of them? Will we have another examples or application that we can use them? Yes.
You still have metagenomics, right?
Excellent. Thanks, Adi. Yeah. So in metagenomic, we don't care about reads, for example. We care about the comparison of genome to genome, right? Yes, we do read to genome in the beginning, but at the end, we try to figure out what is this genome. Even if the sequencing machine will give me the full DNA, I still don't know who is the owner of this because I just swap a sample from train. I have no idea who was touching it, what bacteria, what viruses exist, right? So even if I'm getting the full DNA, that's still not helpful. Even if I know the length, it's not helping me at all. Why is that? Because there are a lot of species, they have the same length of the DNA, especially in bacteria and viruses, all of them almost 1,000, few thousand long. But that does not give me anything about the identity. So I still need hardware accelerator to compare genome to genome. And when you compare genome to genome, it's no way to compare it as one piece. You still need to chunk it, do kind of indexing, sequence alignment to find these differences optimally and so on. So all the things we presented over there, they will never be trashed, or they will never be outdated or whatever you feel about the future of this. And think about also pangenomics, where we compare full genome with full genome of individual species or human, basically. So finally, we came to the end. Sorry if I take more time. System design for bioinformatics is really critical, as you can see. It really has impact on almost everything around us, not only our health, but also the environment around us. And this is apparent from the pandemic we see. The lecture is about accelerating a key step in bioinformatics called genome sequence analysis or read mapping. Many bottlenecks exist in accessing and manipulating a huge amount of genomic data. We were explaining about read mapping, but if you remember, there is a step called base calling in ONT devices that is also time expensive step. That is also a bottleneck. But normally we do this step inside the sequencing machine. That's why I was focusing on read mapping, which is actually the first bottleneck in the genome after base calling. We cover various recent ideas to accelerate read mapping. Hopefully that gives you a motivation. If you are interested in this topic, I'm happy to discuss many of these research ideas with all of you. The key takeaways that population scale analysis are not an easy task. Definitely, scalability is a very important thing, and it's very difficult, very challenging. You need to consider many things in designing a new system. For example, if you are a good hardware designer, you might not be aware about genomics, so you need to learn this topic. Then you need to design an algorithm for that. Then you need to design hardware for that. But that will be so much fun, I would say, based on my 8-year experience on this topic. It will be very rewarding, very impactful at the end when you see someone are adopting your own design and real practical clinical setup. That will be really rewarding at the end. It will enable a great future. It has large scientific and medical implication in everything. Very hot topic these days for graduate studies and research. And for that, I would say most speed up comes again from parallelism gained by algorithm and hardware architecture. Please don't forget that. It's very important. I would like to acknowledge everyone who was participating on all the research I was presenting, all the funding agencies, Professor Onur Mutlu, my PhD advisor, and our great collaborator, Sergey. And if you are interested, if you are part of ETH, interested in doing research, please talk with me. We can propose a project together. We can work on it for FPGA, GPU, SIMD, C implementation, and so on. If you are outside ETH, please use this link to apply to our system. We will be in touch with you as soon as possible. And thank you so much. That was all. The next are just recommended reading. Hopefully you can check some of them if you are interested. There are a bunch of videos as well that you can watch, especially if I didn't cover some part of the lecture fair enough. All these public available with the links. These are papers. You can check all from our group. And thank you so much. I can take question offline if you want to stay. I can stay a couple of minutes. You can send in Moodle. You can send by email as well. Thank you so much for all the great questions. I enjoy it, enjoy answering them. So yeah, take care. And happy to take question now. Yeah, time to have maybe dinner, early dinner. Okay, thank you so much. Yeah, take care. See you next week. Yeah.
I wanted to ask them offline. I don't want to
Okay, let me stop this.
So, firstly, you mentioned the word heuristic many times. Is it safe to basically